good morning, everybody, and welcome. And someone told me since I need no introduction, I'm not going to introduce myself. It's my pleasure to be your moderator today for this incredible day of presentations addressing the evolutionary foundations of human uniqueness. I welcome all of you on behalf of the great Arizona State University, and particularly the Institute of Human Origins. As all of you know, I think we are celebrating our 20th year at this extraordinary university this weekend. A very productive 20 years here, and since we arrived in the summer of 1997, our research agenda has significantly broadened to cover the entire span of human evolution, from the origins of our very earliest progenitor, who split off from a common ancestor with the African apes 8 to 10 million years ago, right on up to the emergence of modern humans, like all of us in this room today. This morning and this afternoon, we're going to hear the results, at least preliminary results, of an intensive 3-year study, including some 60 external collaborators carrying out some 13 thematically linked scientific research projects. And the goal of this research has been to bring us closer to understanding the evolutionary interplay between complex cognition, our reliance on cumulative culture, and our exceptional capacity for cooperation. Today we have assembled an outstanding slate of highly accomplished and internationally respected speakers, all of whom are associated with Arizona State University in one way or another with the Institute of Human Origins. And I can assure you that what you're going to hear today comes from scholars that represent the very best and are not only world-class, but they're exemplary teachers and mentors of the anthropologists of tomorrow. Our first speaker is Bill Kimball, who is director of the Institute of Human Origins and the Virginia Ullman Professor of Natural History and Environment. A little bit incestuous. Bill and I have known each other since early 1970s, and when he first took a course from me at Case Western Reserve University, and then went on to get his PhD at Kent State University. He's been especially focused on paleoanthropology, particularly African paleoanthropology, but has also done extensive work with Neanderthals, particularly in the Middle East. And he has written widely on evolutionary systematics. So today he will speak to us about, was Darwin right about human origins after all? Bill Kimball.
Thank you very much, Don. Is this mic live now?
Yes.
Good. Thank you very much for that introduction, and to the Templeton projects in general, and good morning, everyone. In his 1871 magnum opus, The Descent of Man, Darwin made 2 claims in the absence of a fossil record of human evolution. He suggested famously, first of all, that when the first fossil evidence of human origins would be found, it would be found in Africa, based on the extensive similarities that were understood in his day between the African apes and humans. The second claim that he made involved a complex causal explanation for how we became human. An explanation that invoked bipedal locomotion, enlargement of the brains, complex tool use in the pursuit of a lifestyle dedicated to predation and defense. Now, for many years, since Darwin's day, the accumulated fossil record had suggested that while Darwin was certainly right in his first claim, the timing events related to the second claim was false. We understood in the 1960s and 70s that the earliest record, for example, of enlarged brains and stone tool use was not associated with the earliest humans, where bipedalism long preceded those critical events that he had tied together in a single package. And what I'm going to suggest today is that, in fact, what we now understand about the earliest hominins preceding the genus Homo, the one to which we belong, suggests, in fact, that Darwin may have been more right than wrong. Now, a lot of what I'm going to say today has developed out of a 3-year ferment of research and activity at the Institute of Human Origins that has stemmed from the John Templeton Foundation's investment in our initiative called the Evolutionary Foundations of Human Uniqueness. And over those 3 years, through large group interactions, through one-on-one conversations, a consensus is beginning to emerge about some of the earliest stages of human origins. And what I'm going to tell you today is a product of those interactions that could only have happened in the context of that grant. And I want to particularly recognize Dr. Paul Wasson, whom I cannot see, but I know is out there, Director of the Life Sciences Division at the John Templeton Foundation in Philadelphia, for recognizing early the potential of our group to carry out an audacious research agenda that we had proposed to them back in 2012 for the first time. So thank you, Paul. Now, I need the famous clicker. Turn it on. It's on. It's beeping and vibrating and all kinds of things. All right. Now, since Darwin's day, of course, we have ample evidence, largely from DNA, that not only are we uniquely related to the African apes, but we are uniquely related to the chimpanzee. And one of the tasks we have as human origin scientists is to explain this unique package of adaptations that distinguishes us not only from chimpanzees, but from all other living primates. Our large brains, our diminished fang-like canines, our tiny chewing apparatus, our teeth and our muscles are very small compared to those of other primates relative to our size. Our unique form of bipedal locomotion, our protracted growth, our technology and language, and on and on and on. Darwin's explanation, which ties together, as I suggested, descent from the trees to the ground as the spark of evolutionary change, and natural selection to free the hands from locomotion for other tasks that facilitated upright locomotion, kicked off a positive feedback loop in his mind among canine tooth reduction, enlargement of the brain, terrestrial bipedality, and stone tool manufacture, all of which began to feed back into one another in an ever-progressing loop. So critical was the idea that the difference between ape and man involved the acquisition of stone tools that the renowned British archaeologist Kenneth Oakley in 1959 basically said, if you want to know when man began, just find the earliest stone tools. You don't need any other piece of evidence. And that seemed to come into reality from the ground in 1964, when Louis Leakey and colleagues recognized for the first time the species Homo habilis for largish-brained, small-toothed, apparently stone tool-bearing hominins from Bed 1 Olduvai Gorge dating to about 1.75 million years. And not only did Leakey and his colleagues tie together evidence of stone tools and brains based on the anatomy of the bones coming from Bed 1 at Olduvai, they audaciously undertook a redefinition of the entire genus itself, the one to which we belong, Homo sapiens. They suggested that by comparison to apes and Australopithecus, The pelvis and the hind limbs suggested a fully upright mode of bipedal locomotion, that the thumb was capable of the sort of motion that would facilitate a power and a precision grip necessary for stone tool use. And of course, these fabulous Oldowan stone tools were being discovered in great abundance in those same beds. And that consequently, the brains were significantly larger than what we saw in apes and Australopithecus. And finally, the dentition, small in the back, large in the front, was not that of a specialized vegetarian eater or vegetable eater, but instead would be more consistent with an animal that was omnivorous, incorporating meat into the diet. This was the dividing line between the genus Homo on the threshold of humanity and Australopithecus. They suggested, in fact, that Homo habilis might even have been a hunter, and that other early hominids of the genus Australopithecus found in Olduvai may indeed even have been their prey. Beginning in those days, with the identification of Homo habilis, there became a preoccupation with the idea that Homo habilis was essentially a homunculus, that essential human characteristics that we see today that divide us from non-human species were present in essential form, and that over the several million years since Olduvai Homo habilis, there was a progression, unbroken line of progress in technology, in brains, in canine size, in the perfection of bipedality, the longer period in which we grow, and our sophisticated technology, in an unbroken skein of ancestry and descent. Philip Tobias, writing in 1965, basically said that Homo habilis was the last remaining gap in our story of human origins. It wasn't that simple. Work in the 1980s, led by people like Bernard Wood and Ian Tattersall, who's here in our audience today, and others, showed that the evolution of the genus Homo was not a smooth progression of unbroken ancestry and descent in which all of these adaptations began to emerge into more human-like form over the passage of time. Instead, there was diversity, contemporaneous diversity, especially in the period between around 1.7 and 2 million years, encapsulating the very time of Homo habilis. Now, if we assume, as seems reasonable, given all of these species being attributed to the genus Homo, that they must have shared a common ancestor sometime in the past, we would like to know when that is. When did those species emerge from an ancestor? And the very simple answer is, we don't have any idea. And the reason is this: the time period in which we think the genus Homo might have arose from some Australopithecus-like ancestor is virtually empty for the Homo lineage. That has not stopped scientists from positing climate change-induced environmental degradation after 3 million years ago as the root cause of cognitive advancement, technological sophistication, increasing carnivory, and all manner of adaptive change leading to later Homo, right through a period of time in which there's no fossil evidence. This is the record of the genus Homo between 2 million and 2.5. You could put it in a shoebox and leave room for a pretty good-sized pair of shoes. There's very, very little evidence that bears on the adaptive evolution of the ancestor of species diversity in the genus Homo, which we know existed between 1.7 and 2. Now, the work that Kay Reed and her colleagues have done at the Lady Gararu site in Ethiopia has added a critical new data point to that time period with the finding of the Lady Gararu jaw at 2.8 million, which we attributed together with Kay and her team to the genus Homo. And as we sit here, it is the earliest known fossil of that genus. But it adds just one data point in a time period in which we have very few data points. The question is, if we have the genus Homo at 2.8 million, and if stone tools are so critical to the emergence of the genus Homo, do stone tools extend back that far? Don't know, maybe. Can we drag the record of stone tools back? The 1970s biography of Lucy suggested essentially that she was an upright ape, that she had an ape-like brain size, that her skull was predominantly that of an ape. She did have small canines, and as far as we knew, no stone tools. An upright ape. Now you'll notice here that there is in time an unlinking of characteristics that Darwin thought were critical to the emergence of humans. For example, Lucy was a terrestrial upright biped, but she had, according to this biography, an ape-sized brain. Unlinked. No stone tools. Unlinked to the emergence of bipedality. She had small canines. Unlinked, contrary to Darwin, to stone tools. Then things began to change. And I'm going to suggest that the change that began with the publication of this paper in 2010 has led us to a reevaluation of the evidence for the paleobiology of Lucy and the earliest Australopithecus species. And that reevaluation has basically raised some new and fascinating questions about whether, in fact, Darwin wasn't closer to the truth than we had given him credit for. 2010, a team led by our former postdoc, Zarasinai Elam-Sagan, whom you heard last night, perhaps, speak about his experiences at IHO, participating with Curtis Marion here in IHO, suggested, based on finds from the Dakika site across the Awash River from Lucy site and the same age, that hominins in that time period may actually have been using stone to harvest meat protein in some fashion from medium-sized mammal bones, from medium-sized mammals. Controversial. Controversial. And we have, as part of our Templeton-funded projects, students working with Curtis and with us in Ethiopia who are testing some of the hypotheses that come from these finds about cut-marked bone at 3.3 million years ago that should lay to rest some of the controversy surrounding these claims. Remarkable, though, if true, that the first evidence for the modification of mammal bone by stone tool-wielding hominins predates the genus Homo by some 500,000 years or more, based on the lady jaw that Kay's group found. And we are working through Templeton-funded work with Jessica Thompson and other people working with Curtis on trying to increase knowledge of the sources of bone modification in these early time periods. Out in these deposits in the East African Rift Valley, bone can be marked by all kinds of damage that stem from the time the animal died to the time we find it as a fossil. And one thing we know from the work of Jess out there with us in Hadar in Ethiopia, in the same age beds as the bone fragments that came from Dakika across the river, is that there are numerous sources of bone modification. And one of the most common is that of crocodiles. Hadar was a drive-up window at McDonald's for crocodiles. They are all over the place. And one of the most important tasks that students like Jake Harris and others who are working with Curtis under the Templeton grant are doing are applying very sophisticated morphometric, numerical, quantitative methods to try to create probabilistic models to discriminate various sources of bone modification in the past, and that'll be critical to solving some of the debates that have come as the result of those Dikika discoveries in 2010. Equally, if not more impressive, came from the west side of Lake Turkana in 2013, published a couple of years ago, was the first evidence for crude stone tools that are approximately the same age as the Dikika cut marks. These are cruder, larger, less sophisticated than Oldowan artifacts from Olduvai Gorge that Leakey had access to when Homo habilis was named, but they do seem to be genuine. So, the earliest stone tools, the earliest suggested evidence for bone modification by tool-wielding hominins, all of a sudden pops up in Lucy's time period, well before the genus Homo. The question is, does Homo go back that far? Don't know. But I'm going to suggest that what we now know about Lucy's paleobiology, that she very well may have been the first stone tool maker. First of all, brains. Lucy did not have an ape-sized brain, in fact. We now know that in relation to chimpanzees, the earliest Australopithecus specimens, like Lucy, attributed to A. afarensis, had brains that averaged 25 to 30% larger than those of chimpanzees. And if we assume that that was the size of the brain of the common ancestor, that is a significant increase. 2, the foot. Chimpanzees, you will all know, have a foot with a mobile first toe that resembles our hands. Those feet are used for grasping, climbing, manipulation in the trees, and our feet are rather different. No mobility of the great toe. And in 2011, with our colleague Carol Ward, we published evidence from the Hadar site that the foot of Australopithecus afarensis was endowed with a complex set of arches like our own feet. And what do those arches do? They do 2 things. First, they provide a rigid lever for the foot for flexion of the toes when you step off in a bipedal stride like we do. And second, the arch provides a passive energy storage and release mechanism, all the soft tissues under the arch, that actually has the effect of reducing the cost of locomotion by about 20% when you're walking in a bipedal stride, especially when you're running or when you're engaging in endurance walking over distance. 2, 3, the hand. We've known for a long time that the hand of Lucy, the hand of Australopithecus afarensis, is like a human hand. It's not like a great ape hand. The thumb is long, the fingers are short. Work published by our colleagues Sergio Almathia and David Alba in 2014 showed indisputably that when you look at the digital ratios, the ratios among the thumb and the other digits, they look just like humans, falling into these green bands here with modern humans. And this harkens back, actually, to a paper written by our own emeritus professor of anthropology, Mary Marsky, and 2 IHO grad students in 2008, in which they say the hand of afarensis is likely a reasonable model for the hand structure of hominin species responsible for the earliest, then known, earliest stone tools in 2.5 to 2.6. And 4, the diet. Now, those of you who know a little botany may know that plants differ in their photosynthetic pathways in the efficiency with which carbon is incorporated into the plant tissues. And there's a continuum between what are called C4 plants, which are plants that are highly efficient at incorporating carbon into the tissues in hot, open, sunny environments. And there are C3 plants which are less efficient in incorporating carbon and do best in relatively closed, shaded, cool conditions. Well, it just so happens that when your teeth are forming up in your jaws, they incorporate the isotopes from the food you eat into the enamel. And by taking microscopic little samples from teeth, living or fossil. And through mass spectrometry, we can actually tell what the ratio is and which part of the vegetation community was preferred by the individual when its teeth were forming. And we normally go for the first molars because in humans, at any rate, the first molars begin to form and then erupt around the time of weaning, when animals are moving from mother's milk to solid food. And what you can see here is that at 3.5 million, Australopithecus afarensis, Lucy, is among the first hominins to exhibit an expansion of the dietary niche to incorporate C4 foods, those that grow preferentially in open, high sun environments, compared to early Australopithecus and other hominins before. Now, 4 pieces of evidence. Back in 2000, our own Kim Hill, as part of a team led by Hilliard Kaplan, published an interesting paper in the journal Evolutionary Anthropology, in which, in comparisons of modern human hunter-gatherers and chimpanzees, they posited that at some point in human evolution, there was a switch from a grade 8 preoccupation with leaves and fruits and insects, which require relatively low skill to acquire and have a low nutrient density, to one which humans specialize in, where the density of nutrients is high, but the skill required to acquire them is also high. I would suggest that that shift is not tied to the appearance of the genus Homo. I would suggest that it's tied with the earliest known Australopithecus. And that when you look at the evidence for stone tool use, the evidence from the foot, the evidence from the hand, the evidence of brain size and an eclectic diet, which implies, to my way of thinking, the exploitation of new food niches in open environments, you suddenly come round to a new view of Lucy. Now, we don't actually need chimpanzees to do this exercise because we actually have something that many of us think exemplifies the ancestor to her. Not necessarily the ancestor, but exemplifies the ancestral condition, and that's Ardipithecus, which we know is related, or some of us think is related, to humans by virtue of its part-time bipedality, the form of its skull base, and the size of its canines. If we take that as a given, and we ask then what happened here, the answer is a lot. Ardipithecus, like chimpanzees, has a tiny brain. By comparison, Australopithecus has a very big one, some 30%, 35% bigger. We have a hand of Ardipithecus. It has a short thumb, long fingers. Lucy, long thumb, dexterity. Ardipithecus, like an ape, a divergent big toe, climbed in the trees a lot. No arches in its foot. Lucy, like us, arches. Implies to me an adaptation of habitual terrestrial bipedality, probably associated with expanded foraging niches, and those foraging niches are also given to us by the increased range of variation in the types of plant foods that these animals were eating. The brain, the hand, the foot, the isotopes, the form of the chewing apparatus tells us that Lucy afarensis looks a lot more human-like than we thought. And so, we now have stone tools back with this animal, with its large brain in relation to its ancestor, with a human-like foot in relation to its ancestor, a human-like hand in relation to its ancestor, and an expanded dietary niche. Darwin may have been right, but we need to amend his hypothesis. Out go the canines, because we know Ardipithecus had them, small. Predation and defense, not so likely. We would substitute in there, or I would, extractive foraging, foraging for food items that are either hidden or require some mechanical activity to extract the nutritive portion in varied environments, open environments, closed environments, as suggested by the C3-C4 dietary niche. Increased mobility, dietary flexibility, and enhanced cognition, as we deduce from the increased brain size. If Darwin were sitting out here, he'd be smiling, because it would look familiar to him. And so, the transition, the implication here, two. Number one, an explanatory scheme like this draws together disparate pieces of evidence from early Australopithecus biology that otherwise are mysterious. The increased brain size, the human-like hand, the human-like foot, the increased foraging range as suggested by dietary diversity in those isotopes. And secondly, if this is correct, it also means that the transition from Australopithecus to Homo, which since Leakey's day has been endowed with this virtually mystical transition from non-human to human, is actually chimerical. And that instead, what we see in the emergence of the genus Homo is an intensification of adaptations, behaviors, and so forth that are much more deeply rooted in our ancestry than we had thought. Thank you very much.
Lots of grumbling on the side here. Thank you, Bill, for that certainly provocative presentation. Interesting how over time our perceptions of what we thought we knew begin to morph and change. Also very interesting about that talk, and by the way, we'll be doing questions and answers, hopefully, later in the afternoon. But what this presentation really highlights in so many ways, and this is particularly important for students who are deeply immersed in Darwin's revolutionary idea of evolution and modification through time, is that we hear often at cocktail parties and elsewhere about paradigm shifts. And as we look back, we realize, I remember reading The Structure of Scientific Revolutions when I was a student at the University of Chicago. And a scientific revolution would be the fact that, the Earth goes around the sun, not the other way. And Darwin's view is certainly a paradigmatic shift, a shift totally in the way we view the natural world. And that's one big takeaway message from Bill's presentation. The other takeaway message from Bill's presentation is the absolute stunning brilliance of Darwin, in the sense that Darwin really never had a job. He just created the core of evolutionary biology out in Kent. But he was able to use his observations to bring about this extraordinary shift in our understanding of the biological world. And the theory of evolution is still the core to biological thought today. Darwin wouldn't know many of the things that Bill mentioned. He didn't mention genes, but if I talk about genes and DNA, you all know what I mean. Darwin, in 1859, when he articulated the origin of species, he wouldn't have known what genes were. He didn't know how things were inherited. But he knew that there was a unifying principle for the biological world. And to me, that brilliance is expressed in the sense that this is a total shift in our thinking about the biological world, and it is one that has survived and flourishes today. And the other thing that's interesting, another paradigmatic shift, of course, was Einstein in the early 1900s. But a handful of people in the world would have been able to pick up those articles by Einstein and understand them. Yet when Darwin published his book, The Origin of Species, on November 24th in 1859, and was sold out in one day, any educated person in the world could pick up that book, read it, and understand it. So, at any rate, thank you very much, Bill. One thing that IHO is continuing to do is to try to not just make new discoveries, those make the headlines, but to actually stand back and embrace a much broader transdisciplinary view of how this information allows us to better understand the human career. We've heard about the early part of the record, and indeed our work when we first arrived here was intensely focused on Australopithecines in general, and yet we were interested in other aspects of human origins. And what interests us more than ourselves? You spend most of your life thinking about yourself, and we all belong to Homo sapiens. And we all want to know how, where, why, under what conditions did we emerge? What is certainly emerged within the study of paleoanthropology is that Africa, in every manifestation, is the root of every important transition, species, innovation, and so on in human prehistory. As I wrote many years ago, no matter where you grasp a branch on the human family tree, the roots lead back to Africa. Whether it's a common ancestor to humans and African apes, whether it's Australopithecines, whether it's whatever Homo erectus is, whether it's the first tools, whether it's the first upright walking, whether it's the first brain expansion, whether it's the first out of Africa, 1.8 million years ago, Darwin was correct, as was, and I would like to mention my hero, Thomas Henry Huxley. And our research has really bolstered that, and sort of the overall, overarching message that this research gives to those people out there who want to know something about where they came from is that we are all, in terms of our ancestry, Africans. When Curtis Marion, after finishing his PhD at University of California at Berkeley and moved east to Long Island, and I understand he was very happy to leave for this beautiful weather in the Southwest, joined us as a professor in the School of Human Evolution and Social Change. He's become associate director of the Institute of Human Origins. He occupies a position, a prestigious position known as foundation professor. And he has very, very broad interests. But those interests really circle around trying to understand the origins and emergence of ourselves, Homo sapiens, supposedly wise man. And his work personifies transdisciplinary research, which is really a hallmark of paleoanthropology. He's interested in modern human origins, African Stone Age, paleoanthropology in general, zooarchaeology, taphonomy, paleoecology, present hunter-gatherer populations, grassland and coastal ecosystems, and so on. And his fieldwork has taken him to various places in Africa, Somalia, Kenya, Tanzania, and most importantly, to South Africa, where he directs, quite a mouthful, the South African Coast Paleoclimate, Paleoenvironment, Paleoecology, Paleoanthropology, or SAC-P4, a transdisciplinary research program approach our understanding of modern human origins, climate, and environment. And today he's here to talk to us about Pleistocene coastal South Africa, a special environment for the evolution of our species. Curtis.
Okay, thanks Don for that nice introduction. I appreciate that. we are going to do a big shift in time. So one of the things I thought I would do with my talk is give you an introduction to that time period between the early record that my colleagues were just talking about and the stuff that I'm going to be talking about. It's a huge chunk of time. So I'm going to begin my talk with that introduction, and that's going to kind of tunnel down to the research questions that my team and I have been investigating. And that team is the SACP4 team. I remember when I originally was trying to come up with the name and the acronym, I circulated an email where I suggested for the acronym SAC4P. And somebody pointed out that that sounded like a urine bag. So I changed it. And that includes probably 45 people from around the world. It's a very multidisciplinary international project, and I'm going to point to some of those people throughout the talk. But there's also a team, kind of a sub-team of that called the Paleoscape team, and I'm going to explain to you in the talk what they actually do and what we're trying to accomplish with that. So I'm going to tunnel down into the research questions, and then I'm going to focus specifically on what we were trying to accomplish with the Templeton grant. And that will bring us to the end of the talk. So to bring you up to speed on the material that we're working on, I'm going to talk a little bit about the origins of modern humans, and that will set the basic research questions. And at the bottom of this graph, I've put a proxy for world temperature, and that is the delta deuterium record from the Epoca ice core, so that's the Antarctic ice cores, and it goes just about to 800,000 years ago. They've pushed it back a little bit further now. And when the take-home message from that graph is, and that's in thousands of years, when that line is low, and it's kind of in that blue zone, the world is in a glacial phase. And when that line rises up, and it's in that kind of reddish, orangey color, the world is in an interglacial phase. And of course, we're in an interglacial phase today. And one of the things that you'll note is as you go through time, the amplitude and climate change, or the amplitude and temperature change, gets more and more dramatic. But most of the time of modern human origins, the world is in a glacial phase. So the climate was nothing like it is today, which means that we need to do a lot of work to try to reconstruct what that environment looked like, which is one of the goals of our Paleoscape approach. And then above, I've really simplified the evolutionary record for modern humans into two main lineages, the African archaic lineage and the European archaic lineage. And I'm going to show you a lot of lines, and when they end in a circle, that means they end in extinction. And when there's an arrow, that means it's continuing. So they share a last common ancestor sometime about 800,000 years ago. And there's good evidence to suggest that up until about 400,000 years ago, there's quite a bit of gene flow between those two populations in those major continental areas. And that's probably because people are flowing in and out of Africa and Eurasia pretty easily. And then sometime around 400,000 years ago or thereabouts, that gene flow gets cut, and that's probably, at least in my opinion, is probably due to the formation, the maturity of the Saharan ecosystem, stretching all the way across northern Africa into the Arabian Peninsula, which cuts off or makes very, very difficult that gene flow. So then you have to have really strong interglacials to create a grassland in that Sinai-Negev passageway so populations can get in and out. And of course, when that gene flow gets cut, those populations start to diverge. We have a lineage called the Denisovans. I'm not going to get into it in much detail, but they appear, and they were documented from a cave site in the Altai Mountains in Russia, from a finger bone that I think was a finger bone that they first thought was a Neanderthal, and it turned out to be an entirely new lineage. And then all of you have heard of Neanderthals before. We know a lot about the Neanderthal lineage. Of course, both of those end in extinction. And then the modern human lineage and the fossil record and the genetic record seem to be consistent about this, appears probably around 200,000 to 150,000 years ago. And of course, everybody on the planet today is descended from that lineage. And then around 70,000 years ago, a very, very small founder population of that lineage leaves Africa, and I think probably through the Sinai-Negev passageway. And that individual, in fact, there's a fossil from Israel now that actually seems to date exactly to that founder population's time. I mean, that could be one of the founder populations in that cave. They seem to leave Africa about 70,000 years ago, and those end up being the two major populations, Eurasians and Africans. And interestingly, as that founder population exits out of Africa, of course, it encounters the other hominins that are in the world at that time, and they interbreed with Neanderthals such that all Eurasians have a small amount of Neanderthal DNA in them. And then there's this spectacular radiation of those modern Africans and Eurasians into all of the ethnolinguistic groups that we see around the world today. And a subpopulation of those modern humans moving southeast into Southeastern Asia encounters Denisovans, interbreeds with them, and picks up some of their genes, disperses into Papua New Guinea and Australia such that the indigenous inhabitants of Papua New Guinea and Australia still have some of that Denisovan DNA in them. There's an interbreeding event with an unknown archaic African hominin and modern Africans. So the take-home story there, of course, is while we don't have any of these recent hominids left living with us, we have bits and pieces of them in our genome. And it would be great to figure out what those bits and pieces do. And we will eventually. But one of the points I want to make is that modern human lineage appears during what is the longest, coldest glacial phase in the last million years, something we call marine isotope stage 6. And it dates from about 195,000 years ago to 130,000 years ago. So the world was locked in a cold glacial phase. And Africa normally responds to glacial phases by getting very, very dry. So for example, the Sahara roughly doubles in size during one of these intense glacial phases. And the Kalahari-Namib Desert grow together, and they also expand. And what some of us have argued is that probably creates a situation, an environmental situation, where you have maybe 4 to 6 localities in Africa that would have been good refuge zones for the population that ends up being the founder population of modern humans. And I've argued for a variety of reasons that the likely candidate zone, the best candidate zone, is the Southern African subregion. So that's why my field project is there. And that argument was put forth in a scientific paper that I wrote for the Journal of Human Evolution, but was also published. I wrote a general public science version for Scientific American. And the hypothesis is basically an ecological potentiality hypothesis. And without getting it into too much detail, you can go and read those papers, whichever one you want. I argued the reason is that you have one of the richest intertidal zones anywhere in the world, providing a very rich source of protein, very high quality protein, combined with an extreme diversity of below ground tuberous plants. And that results from the special winter rainfall system that you have in the Cape. So you put the two together and you have this outstanding diet of protein and carbohydrate, but importantly, both are very resilient to colder temperatures. In fact, intertidal zone productivity goes up when ocean temperatures get colder, as opposed to most terrestrial environments where the productivity goes down. So we've targeted the southern coast of South Africa, and my team has been doing 17 years of field work. And I've put in red the records that are the result of our field work, most of it at Pinnacle Point, which is in the numbers PP13B, PP9B, et cetera, Fleerspy is a new open-air locality that we're working at. And I've compared those records to the other major sites in South Africa in yellow. And you can see that one of the things that we've been successful at, that we've really been slogging away at year after year after year, is developing a very long, complete sequence. But you can see we still have some gaps that we need to fill in, and our team will continue that field work until every one of those gaps is closed. Now, one of the things that we've learned from all of this work is that the South African coastal record for modern human origins just looks different than any other record you find any other place in the world. So different from what we see the Neanderthals producing, or even other areas in Africa. It's richer in material cultural complexity and has different things happening. So the question then becomes, why is that? But let me quickly review what some of those characteristics are. And some of our work has contributed to this. So in 2007, we published a paper in Nature which documented the earliest evidence for people expanding their diet to coastal resource use 160,000 years ago. And that's still the earliest record that we have. Has the earliest record for modified pigments, which are on your left-hand side on this. And that was also at PP13b, dated to 160,000 years ago. The earliest evidence for complex use of fire, what we would call pyrotechnology, using heat to transform the material properties of, in this case, stone, so that you can improve its flakability. And of course, pyrotechnology is the foundation of our technology, but this is probably the bridging technology to ceramics and to production of metals. Again, it's 160,000 years ago at Pinnacle Point. Some of the earliest evidence for symbolic activity. These are seashells that we find in the record, and we know that they're seashells because the animals were collected when they were dead. They have beachwear on them, and that only comes from when the animal is dead and brought into the site. The earliest evidence for engraved ochre comes from coastal South Africa. This is from Blombos Cave and Klein Kliphuis. The earliest evidence for beads come from coastal South Africa, from Blombos Cave. The earliest microlithic technology, which is an advanced high-velocity projectile technology, we published that in Nature in 2012. It's from Pinnacle Point, also from coast of South Africa. And the earliest evidence for canteens, water-carrying technology, and decorated water containers comes from coastal South Africa. So why this unique character? One hypothesis. This is the origin population. This is the first modern humans that are cognitively complex. And that's what I suggested in those first two papers that I wrote back in 2010. But there's an alternative hypothesis, and I'll just say that it would be the human-environment interaction resulted in this complexity, that we had complex cognition and socially networked hominids throughout Africa, but there's something about the richness of this habitat that stimulated that material cultural complexity. And that's a second hypothesis, and I put that one out in these two papers, 2016 and Philosophical Transactions, and I wrote a Scientific American paper that was published in 2015 for the same idea. So let's go back to this issue of the fact that the climate is very, very different. If we're going to test that hypothesis or probe that hypothesis that it's the environment and human interaction that's driving that complexity, we have to reconstruct that environment. What was it like? That's a big job. So one of the primary goals of the Templeton proposal was to take theory and link it to the paleoanthropological data. And the theory that we've rested on is behavioral ecological theory. And Kim Hill, I recruited Kim Hill to the project to lead that work. And he's going to talk later and talk right after me in more detail about what he's done and define for you what behavioral ecological theory is. And of course, we're taking the behavioral ecological theory and we're linking it to the South African data. And there's so many people in this project to mention, I can't go through them all, but I'll just point out my Norwegian PhD student over there with the weird glasses. That's Simen Ostmo. He developed an agent-based model grounded in behavioral ecological theory to understand stone tool use. He's defending his PhD on Tuesday at 9 a.m. if you're interested. Next to him is Irene Alama Esteban. She did her PhD on our work, just defended her PhD at University of Barcelona in December. Fabulous person working on phytoliths. Ben Scoville, who did his PhD here. And I just want to give a nod to my fabulous colleague in South Africa, Richard Cowling, who's one of the world's most prominent botanists and also a fabulous surfer, even in his mid-60s. Unbelievable. And the way we implement that behavioral ecological theory is through climate, vegetation, and agent-based modeling. The vegetation modeling led by Janet Franklin here at ASU, and recent PhD Alistair Potts way at the other end over there. But what I'm really going to talk about is the agent-based model, and that work is led by Marco Jansen, who's a professor here at ASU, and Colin Wren, who was a Templeton postdoc, and no sooner did we hire him as a postdoc, and he gets a job at University of Colorado, but he kept his leadership of the work. So what we argued in a paper published in 2015 in the journal Evolutionary Anthropology is that the way to approach this is through building something we call a Paleoscape model. And what we mean by that is a temporally dynamic computer model that projects onto the landscape all of those resources that are important to a hunter-gatherer in regards to their return rate to the hunter and gatherer. And then we use that to create quantified hypotheses of human behavior. So we have the computer model generate what we think people should be doing. And our goal is to take paleosciences and its reliance kind of on informal models, and I'll make a distinction between informal and formal in a second, from a reliance on informal models to these kinds of formal models. So we know climate change was massive in the past, rotating between these glacial and interglacial stages. So what we need to do is we need to take models and project what those resources were like under these wildly different climate conditions. And that is really, really hard to do. So for example, here's an informal model on your left-hand side. We know that during glacial phases, when seawater went down, there was a massive plain exposed off the coast of South Africa. So if you were in one of the caves that we're digging today, sometimes there was a plain 90 kilometers wide. Looked very, very, very different. So a picture like this is your informal model, but we tasked Eric Fisher, who is also supported by the Templeton Grant, to develop a quantitative model. So he built a computer model that shows where that coastline is at any given slice in time. And that's the output of the model. So I'll just point out that, I know I'm going to do this wrong. No, good. Here's your distance in kilometers. 138,000 years ago, the coast was 95 kilometers away. So that's the output of that computer model. Formal model, right? It's telling us exactly where the coast was at any given time. That's what we need. And then we enrich that model by putting the vegetation on it and the rainfall on it. And Kirsten Braun, who was a Templeton postdoc, is leading the study of the stalagmites, isotope analysis, and so on. And that's one of the proxies we use for figuring out how much it rained in the past and what the vegetation was like. And then Haley Cawthra, who is a South African, did a PhD under us. She's a marine geophysicist, and she mapped the offshore rocks. And that also helps us put the topography and the vegetation, potential vegetation, on that offshore platform. And Sandy Copeland studied the isotopes, the strontium isotopes of the antelope. And what she was able to find was that all of the antelope that were hunted and their bones ended up in the caves lived on this exposed continental shelf when the people were living there. So what we've hypothesized is that in the past, there was a migration ecosystem of the animals moving east and west. The reason being is that in the east you have summer rainfall, in the west you have winter rainfall, and that provides the push-me-pull-you to set up that migration ecosystem, because the animals are always chasing the green grass. Right? And again, the goal is to build a formal model. How many animals would be there? When would they move? So that we can use that to predict what the hunter-gatherers would be doing. So here's an example of our informal model, right? During warm climates, the coast is nearby. We would have mostly winter rain. We figured that out from the stalagmites. You'd have C3 grasses and a more western flora. And then during colder climates, you have a plain here and animals are running back and forth, and the coast is far away. We have mostly summer rain, and again, Kirsten and our colleagues, Mirabar Matthew, figured that out from the stalagmites, and mostly C4 grasses and so on. But how do we take that and turn it into strong hypotheses of how hunter-gatherers would behave? And how do we build this formal model? Well, what we do is the way we did it was we created habitat types. Okay, so here's the Mosel Bay area, and each one of these is a habitat. And then we had modern humans that live in that area and are familiar with the vegetation and the animals and with the intertidal zone, experimentally forage the foods in those habitats. So for example, that was studied by Suzanne Butte, who is a PhD student associated with our project at Nelson Mandela University. And then we had coastal people experimentally forage the coastal resources. And that was studied by John Templeton, who was also a PhD student at Nelson Mandela University and affiliated with our Templeton grant. So our overall team for building this, I just want to give a shout out to all of them. The South African CSIR, they were responsible for the climate modeling. Nelson Mandela University, climate modeling, vegetation modeling, and foraging studies. Stellenbosch also contributed to the foraging studies. ASU was the leader on this, and the Supercomputer Center in Pittsburgh ran the supercomputer modeling study of using the regional South African climate model, which we ported to those supercomputers. What can we do with this model? So years ago, I put forth a hypothesis that effective coastal foraging is cognitively challenging. You can't just wander out on the landscape and be an effective coastal forager. If you do that in South Africa, you'll get killed in the intertidal zone. It's rough. And the reason is that when the moon and the sun are not in alignment, you have something called a neap tide. And those of you who know the coast know this already. But in a neap tide, the high and low tide variation is very small. But when the moon and the sun are in alignment, so you're in a new moon or a full moon, you have what's called a spring tide. And spring has nothing to do with the season. It means it springs back wildly, back and forth. So low tide is super low, and high tide is super high. Right? If you're a good fisherman, the time you want to be fishing there is as the tide is pulling out during a spring tide, and you're following it out, right? And then you're throwing your line in right into the richest part, and then you get the hell out of there, because within 2 hours, the tide comes back up at a spring tide. But the important thing is that when you're at low spring tide, you have this enormous area of the intertidal zone. It is just unbelievably productive, and you can just clean it out and get thousands of calories within a short moment. But the thing to remember is this is running on the lunar schedule, not the solar schedule. So you have to understand the relationship between the moon and the tides and the timing to be an effective coastal forager. So that was the hypothesis. So here's our agent-based model. And Colin put this together for me. So and it's loaded with all the return rates from foraging for coastal resources collected by Jan and directed by Kim. So the idea is that if you haven't figured out the relationship, you're not cognitively complex to build a system to understand and communicate through social learning. This system, the relationship between the lunar system and the tides, you're not going to be able to be an effective coastal forager. But if you're smart, and you can figure it out, and you can build a calendar, right? A calendar is a complex symbolic system. Have any of you ever gone to the coast and bought the tidal calendar? Holy crap, it's like the Mesoamerican, sun gauge thing. Because the tides change at 50 minutes a day, not 60, right? It's really hard to get there at the right surfing time. Okay, so we can run the model. I hope this works. There it goes, Pac-Man, right? There's the agents, and they're foraging across the landscape. And over here, what it's doing is it's outputting the daily output of the net return rate. So we're running an experiment with our agent-based model. Okay, so let's kick out and see what the result was. Here's when you're a dumb coastal forager, and you haven't figured out, so we're removing the cognition, right, to figure that out. That's when they arrive at the coast, and there's your return rate. So this is your net return rate here. This is the days, right? They hit the coast, and they have a fairly flat return rate. This is the output of one of these simulations. And here it is when we get Einstein, and he's figured out the relationship between the moon and the tides, and look at that return rate. They're hitting those spring tide moments, and it's demolishing the return rate for the dumb coastal forager. So the model becomes a tool that we can use to, within the parameters of the model, test that hypothesis, but explore different possibilities. Adding cognition, we could add social learning to the forager. And this creates an understanding how the system operates, but quantitative hypotheses that we can test with the archaeological record. So why is this character of the South African archaeological record so unique? Why is it rich? Well, maybe it's because this was the origin population. But it could also be because the human-environment interaction there just resulted in that complexity. So I'm just going to leave you with a final hypothesis. And that'll be the end of the talk. We can get some lunch. It's a hypothesis, right? What I argue is that when the human lineage got smart, one of the first things it started to do was construct symbolic systems of time, which helped it forage better. And one of those on the coast was the relationship between the moon and the tides, and how to schedule your visits to the coast. That opened up the marine diet. And this is my favorite lunch. I always go to Maine every summer, and clams with butter. Awesome. In the Pleistocene, when we have glacial conditions, a huge coastal plain opened up off the coast of South Africa. It added a landmass the size of Ireland, an incredible biogeographic change. And when that happens, we now add to the diet large game. These large game are not present in the Cape Floral region today. The plant foods there do not support grazing antelope. So when you have the Pleistocene and you have that cognition, you've got an incredible, rich, dense protein food resource. But what the Cape Floral region does have an abundance in these interior areas are these below-ground tuberous plants. Now, one of the things we've been doing in South Africa with the experimental collected plant foods is we've been having these parties where we get the plant foods and we cook them up and eat them. And it turned out at one of the parties that one of these was kind of vaguely hallucinogenic. So I came up with the hypothesis that night, so that, and I named it a Shangri-La moment. And that's another hypothesis for why we have this special character, the South African record. So I want to acknowledge my fabulous team in South Africa. I have, over the years, I've built a team of 9 permanent employees. None of them knew anything about archaeology before we got started. Most of them I hired as, in South Africa, what they call casual laborers. These are people you pick off the street at a stoplight. And they're the best excavation team anywhere in the world. Unbelievable. I want to thank the scientists in the project, the permitting agencies, the Mossel Bay community, which is just an incredibly supportive community. That we have a house at the Diaz Museum, which is given to us for free for our project, and of course our funding agencies, the Templeton Foundation, and so on. Okay, thank you.
Thank you very much, Curtis. Left us with food for thought, and now we're going to have food for our souls. We're going to have a lunch break, and let's reconvene at 1 o'clock for this afternoon's paper, and thank you so much for sitting through this morning. Welcome back from lunch. I hope you're all satiated, didn't overeat, and don't fall asleep. We're moving up the sort of evolutionary ladder and looking at human uniqueness this afternoon. Our first speaker is Professor Kim Hill, who is professor in the School of Human Evolution and Social Change and an affiliate of the Institute of Human Origins. He's played a very critical role in the pursuit of the evolutionary foundations of human uniqueness, made possible for us by the generous Templeton Foundation grant. And Kim has undertaken extensive field research in South America, for example, largely in Paraguay, but in Peru and other places, as well as the Philippines. And Kim is particularly interested in hunter-gatherer evolutionary ecology. And as you heard in Curtis's talk, he is one of the articulators of behavioral ecological theory. He's also interested in the evolution of human sociality, social and cultural evolution. And this afternoon, he's going to be talking to us about human uniqueness, what it is, and so on, foraging and cooperation in marine ecologies. Kim.
Sound here. Okay, good. All right, I believe I have sound. So what I want to do is start by building off of Curtis's talk and show you how we go from more general theoretical ideas to building in and acquiring all the details that are actually required to test specific models and specific hypotheses about foraging and feeding and economic behavior. And then hopefully I'll provide the transition little by little to the remaining 3 talks this afternoon, which is making the move from understanding economic and ecological constraints and foraging behavior to really interesting and important social behavior, which really, is the crux of the issue of how humans became so unique with spectacular cooperation, cumulative culture, and so on. All right, so let me see if I can get these to work. All right, so as Curtis mentioned before, we know some things about important history of Homo sapiens. So early Homo sapiens made a lot of evidence that Curtis presented, extensive use of marine resources very early on, not only in coastal South Africa, but then later as Homo sapiens expand out of Africa, there's good reason to believe that they expanded along the coastlines of South and Southeast Island Asia, all the way to Australia in a very short time period, and then a little bit later in human history, expanding along the coastline to the Americas and all the way down to the tip of South America, probably less than 1,000 years, from entering the New World. So I spent my whole life working with inland terrestrial hunter-gatherers, looking at a whole series of questions having to do with ecology, behavior, demography, life history patterns, and so on. And when we started working on what would be the implications of living in a marine foraging niche, we quickly discovered that while there is some interesting, seminal, and important quantitative foraging information, there's much, much less known about marine coastal foraging adaptations than there is about terrestrial groups. So we decided that for a lot of purposes of our modeling, we would have to go out and collect the information ourselves. And for this Templeton Foundation project, I pretty much focused on collecting the required information in marine foraging groups in several places around the world. So the big issues that we're going after, it takes a lot of tedious field work and getting very detailed information that you would consider to be economic information in order to fully document what foraging economies would have looked like in the past. But the big goal really is, what are the implications of the marine adaptation for understanding the evolution of other things like cooperation, culture, cognition, the worldwide expansion and replacement of at least 5 other species of hominins, and so on. And as Curtis mentioned, we do all of this work from a theoretical perspective that's often labeled behavioral ecology, where we presume consistently with other biological studies of biological behavior of other non-living organisms, we assume that individuals adapt to respond to ecological payoffs and constraints most effectively to reach goals that ultimately are related to biological fitness. So that allows us to create very specific quantitative optimization and maximization models about what kinds of things would lead to achieving these goals most efficiently. All right, so this project 4 that I did with Templeton actually consisted of 5 pieces, and the ones in blue, I'm going to show you some of our results. The 2 in black, we're still working on analysis, and I felt like I should concentrate on things we know more about. So we built a computer simulation model of foraging in ancient South African landscapes, and I'll show you a little bit about how difficult and interesting it is to get the details that are required to go, the parameters that have to go into an agent-based computer simulation model. As Curtis mentioned before, and he and I have discussed many times, we're particularly interested in whether or not marine adaptation is essentially the exploitation of dense, predictable, and non-depleting resources. Because when you have dense, predictable, and non-depleting resource base, general behavioral biology has a set of models predicting high levels of territoriality, extensive between-group competition, and this graph here, which comes from a really great recent study in experimental economics, shows that whenever you introduce strong between-group competition, you get much higher levels of within-group cooperation. So here we have a measure of cooperation in an economic experiment. In the original 10 rounds of the game, you see medium levels of cooperation, and then right here, they introduce a second group competing with the first, and you see huge increases in cooperation once you get between group competition. And this is the same experiment done in reverse. So first you have 2 groups competing, and then you take 1 group away, you see big drops in cooperation. So there's really good reason to believe that strong between-group competition could drive spectacular within-group cooperation, which is one of the things we're interested in. Then I'm going to show a little bit about testing assumptions about foraging, whether or not foragers are really just out on the landscape looking for food, or whether or not there's other things that are important, like I'm going to explain what this means to you, the signal value of acquiring game rather than simply the food value of acquiring game. And then I'm going to show you a little bit of my work in Mindanao in the southern Philippines, looking at what happens after food is produced, essentially documenting the cooperative patterns of the socio-reproductive system that includes spectacular levels of transfers between households and between unrelated individuals, in virtually any kind of resources that they're producing over time. And the final thing that I'm working on in Project 4, but I'm not going to show any of the data today, is I'll cheat a little bit and tell you that we already kind of know what the answer is. The interaction networks in marine societies are much wider and geographically extensive than we see in terrestrial societies. And the implications of that for cumulative culture, being able to borrow ideas from thousands of kilometers away if you're in a marine economy versus terrestrial people who never go more than 50 or 60 kilometers from the place of their birth, is that you get much faster and possibly much more extensive cultural evolution along coastlines and so on. So let's start with the computer simulation stuff first. You know, our idea here basically, Curtis went over this a little bit, I'll just show you a little more detail. We have to populate the landscape. So we have agents in our simulation model and they move through a landscape. You have to be able to assign to each cell in a landscape, and in our particular models, each cell represents 1 hectare. It's 100 meters by 100 meters. What would happen if this agent enters into this cell? And what we're trying to establish is how much food would there be there, how long would it take to get it, and what kinds of food are we talking about. So here you see a bunch of hypothetical cells that if this agent moved down here to the coastline, there's 3,300 calories of marine invertebrates. It would take a half an hour to harvest, but instead if they move over here, they can collect tubers. They could get 1,200 calories. It would take them 1.6 hours to harvest. Of course, these numbers are made up, and so the real trick in doing a computer simulation is coming up with these numbers. Turns out to be way, way, way more work than building a model. A good computer programmer can build this model in NetLogo in a day or two, and then what you're going to see here very quickly in my summary is that filling in these numbers takes several years of experimental data collection. So this is really the hard part. So this is what we do. We start with an area of known habitat types. So this is in fact the area around Curtis's cave site here in Pinnacle Point. This is the South African region. We've got different types of habitats. And basically what we'd like to be able to say is if you had an agent, a forager, an ancient human hunter-gatherer on this landscape, and they move into this habitat or this one or this one or this one, what could they do? What are the resource densities they would encounter? How much time would it take them to harvest, and how much food would they get back? Those are the parameters we have to stick into an agent-based model. And when we do that, we end up producing something that looks like this. This is a schematic color-coded diagram that is based, in this case, on the caloric return rate that a forager could get in some regions versus others versus others. So how do we produce numbers that go in? And in a minute, I'll show you an actual agent-based model for a second. How do we produce numbers that go into these? We did 3 things in this project for the Templeton. One is experimental marine foraging. The other is experimental plant extraction. Both of those 2 follow pretty much the same methodological guideline that you take people out to a habitat type, you turn them loose, you follow them for a few hours, you see what they can collect. These are going to be native, locally well-informed populations that know how to collect plants and so on, and we simply make measurements of what they get back and how much time and what species and so on. With the prey populations, so this is for plants and marine invertebrates. When we talk about animal prey, mobile large vertebrates, it's a little bit more complicated because you can't really run experimental hunting project the way we don't have permission to go anywhere we want and turn people loose with ancient bows and arrows and let them hunt down impalas and bush bucks and whatever and measure what happens. So we have a different method for getting at this, where we essentially take observed real hunting data from other groups around the globe, particularly in Africa, and we build essentially a model, a statistical model that would predict what would happen if you ran into prey species with particular characteristics in these areas, even though we're not going to be allowed to experimentally go hunt in those areas. Okay, so plant collection, experimental plant collection, I told you, is straightforward. You take people out, you turn them loose, you weigh everything they get, you take it to a laboratory, you do a nutrient analysis, You time how much time it takes. You stratify this across the different habitat types, record all the different resource types that are there. One of the things we found, I'll just mention one of the outcomes, lots and lots of plants. This is Prionium. It's a freshwater kind of swamp growing plant, and when you collect it, the edible portion looks like this. I don't really know how to describe this, except it's a little bit like a palm heart if you've ever eaten one of those, or maybe the heart of an artichoke or something. Very nutritious. Here's the return rate that you could get from collecting that in kilocalories per hour. 2,600 basically calories per hour. And we know how many plants there are in a hectare and so on, so we can start building economic parameters. I want you to remember that number in a minute because we're going to compare that to marine foraging. So the next thing we want to know is what could they get if they were foraging in the intertidal region. Same thing, we take Khoisan descendant foragers out, turn them loose, they know exactly what they're doing, they're very talented and knowledgeable, and we do the same thing. We time how much time it takes them, we measure their harvest, we sampled the entire coastline pretty much from Bloombos Cave to Pinnacle Point Cave, two really famous archaeological sites, lots and lots of sampling through 2 years of field sites, stratified on 5 different habitat types, so we can see the return rate on alieonite versus hard rock sandstone, Table Mountain sandstone, and so on. These are our team of people. They collect things like octopi and shellfish and so on. In fact, here's a nice little picture that shows some of the major prey items. This one right here, giant turban shells, these are big giant snails, is by far and away the most important thing they harvest along this whole coast. In fact, if you look at this pie chart, you'll see that turbo alone makes up more than 50% of the total calories that were harvested during these experimental foraging bouts. Anyways, remember the main purpose here is to be able to assign different habitats, different return rates. So here I have 4 different coastal habitats, and these are the experimentally measured gain rates that a forager could get by moving into that area and collecting marine invertebrates for a period of time. The other thing, so there's a lot missing from this slide, so something happened, but this is there should be a curve showing you this decrease. This is the return rate as a function of spring tidal level. So lowest spring tides, extremely high return rates, over 3,000 calories an hour, and these are the neap tides. The water level's much higher, and you see really low return rates, about a 10-fold drop, depending on whether you forage at the best tides or at really average, mediocre neap tides. And remember, there's my magic line for Prionium. So this is a very high return plant item, but in the best tides, some of the foragers can get even higher return rates from marine foraging than they can from Prionium. What's missing on here, there were some lines showing what is the average return rate for hunting by bow and arrow hunters in Africa. It's about right down here, showing that there are conditions under which collecting marine invertebrates is better than hunting. Root collecting by Hadza women in Tanzania is way down here. So certainly when the conditions are right, marine intertidal foraging can be preferred even over hunting and certainly over plant collecting. Oh, here we go, it's all coming up now. All right, well, this is just what I described to you. So you can see that the bottom line here, and we published this in a couple of papers, is that under really bad conditions, the marine intertidal zone is not very attractive. Under the right conditions, our best conditions, over 8,000 calories per hour. Let's see again. Let's see if it's going to come up like it did before. Yeah, okay. So these are worst possible conditions. Young men on alieonite, weather condition 1, which means really nice calm weather, no wind, and extremely low tides, spectacularly high return rates. Okay, another thing we needed to know for the model is how much depletion there is over time. If you keep harvesting over and over and over again, should we really allow the model to keep allowing them to get the same return rate? And so we decided to measure this, and Jan de Vink and I staked out plots. These are 300-meter plot squares. You can't really see the markers, but they were there through an entire year, little metal posts pounded into the sand here. These guys are going back every single spring tide and harvesting over and over and over again. So we can measure return rate, and here's another plot on alienite. We can measure what happened over, this is total number of harvests, and remember, you can harvest about once every 2 weeks, so 26 harvests would represent a year. This is a little bit less than a year's worth of harvesting every 2 weeks, continually in the same plots. And you can see the return rate does decrease. This is just raw data, and this is the data statistically corrected for effects of weather and tide and so on. The bottom line is that there is some depletion, kind of steady depletion down to about 50% of our starting point after about a year. On the other hand, there was a lot of statistical noise in our model, and the confidence intervals around these slopes are extremely wide, so that unfortunately one of the things we would have to admit, this is the way science works, everything doesn't always work perfectly. At the end of a year's worth of experimentation, we could say that we actually are not 100% positive whether there's almost no depletion or whether there's very steep depletion, because all those things are within the confidence intervals of our statistical model. But in any case, the best presumption as of right now today, if I were to build a computer simulation model, is I would presume that this is what depletion is going to look like over a year of time. Okay, then we have this complication of how we're going to put game onto the landscape. And this was done by Jake Harris, who's building a model. It's still not complete. And remember I said we can't go out and experimentally hunt, so we've got to go out and find hunters. High-resolution focal follows of Hadza hunters. Jake did 94 follows in the last 2 years. In conglomeration with other information from other hunter-gatherers around Africa, total of 525 total observed game pursuits. We then build a really ugly mathematical Bayesian model to predict what return rates would be for game species that have not yet been observed. So what we're really interested in are two parameters. What is the probability that a pursuit would end up in success? In other words, if you start to try to hunt an antelope, what's the probability that you'll actually kill it? And how much time is it going to take you? And along with already knowing the size and some other things, we can then predict the profitability. And notice that this is going to be a statistical model based on observed data in order to predict extinct species A, extinct species B, and so on and so on. So our ultimate goal is to be able to project back and say what would happen in a hunting encounter between human foragers in, the Paleolithic, in periods of time when there were species that we can't even measure today and observe. So we put all this together, and we build an agent-based model, and I think Curtis showed you one. Here's a starting point. We've got some agents up here. We have 3 camps. After a certain number of days, you can see the camps have moved down to the coast. That's because this happens to be on a low spring tide day. And then on other days, they start moving inland. The brown areas are areas of plant foods that have become depleted and have no more food. And little by little, the camps start moving inland as they deplete all the good plant resources around the coast. Anyways, we're just in the beginning stages of starting to build these models and simulate what ancient economies would have looked like. I need to ask what time I started, and I've lost. Did we start 5 minutes late?
5 minutes late.
Okay, got it. All right, so our conclusion is that intertidal resources can be really attractive. It's really important to be able to predict tides, as Curtis mentioned. If you can predict the tides, you can get spectacularly good return rates. If you weren't predicting tides, you'd be wasting your time down the coast. We can calculate things like how many people could live on this landscape based on total number of calories that were harvested and the depletion and so on. I calculated about 20 adults could live along a 10-kilometer stretch of coast, exploiting marine resources for 10 days per month, providing virtually all of their food on those 10 days. But we still have the problem that for the other 20 days of the month, they would have to be eating something else, which means we really need to focus on the plants and terrestrial vertebrates as well. Okay, the next project is, in order to build a computer simulation, one of the things you have to assume is what is the goal of a forager. And in most of behavioral ecology, we assume that foragers are designed to maximize net energy gain rate per unit time. If you make that prediction, you can then predict what they would do. But starting the 1990s in anthropology, there was a huge debate about whether or not that assumption is empirically valid. And particularly my own PhD advisor, Kristen Hawkes, suggested in a paper in 1991 what she called the show-off hypothesis, that men in particular are much more interested in getting, by their hunting activity, in getting prey that would somehow signal things like good health, good strength, talent, good genes, and so on, so that men are mainly interested in signaling rather than the actual amount of food that they produce. And this led to a huge set of papers which has gone on and on, debating whether or not the show-off model is a better model for men's foraging activity than is an economic energy gain model. So my student Andrew Bishop and I decided that we could go back to a hunter-gatherer population that's really well documented, the Aceh of Paraguay, and actually test the show-off model and find out how people react. So Andrew went with me a couple years to the Aceh in Paraguay. He ran over 19,900, that's almost 20,000 dyadic comparisons where he's sitting down with informants and saying, this or this, which one is better? Which one do you choose, this or this? We had a bunch of prey items. We had hypothetical imaginary hunters, and we would set up scenarios in order to determine, for example, which would provide the most food. Which provides the most food is really straightforward. You can just weigh them, and the biomass is a direct measure. On the other hand, which one is a stronger signal that you're spectacularly strong or healthy or impressive or whatever? Killing a deer or killing a giant anaconda? Or killing a peccary or killing a jaguar? So we realized that sometimes if men are strategizing in order to impress people, to show off and signal, they might be engaging in a very different foraging pattern than if they're trying to get food. So Andrew set up all these amazing ranking tasks. The first one was just simply to rank all of those prey species. Body size we already knew from weights. How hard is it to find and how hard is it to kill? Because we realized that really what people are paying attention to when they're impressed by you being strong is you have to show that you can kill an animal that's hard to kill, or you can find an animal that's hard to find. So we got rankings for all the species, not only in terms of food value, but what we could call signal value, 2 different measures of signal value. It turns out that there are strong intercorrelations between these 3. They're a bit collinear because big things both produce more food and are more impressive. So we were concerned that basically anytime people hunted something big, we wouldn't be able to distinguish between the hypothesis that they're doing it in order to get food for their family or they're doing it in order to impress people back at camp. On the other hand, so killing big animals provides both more food and higher signal, but what if you kill a whole bunch of small stuff versus one big one? All the small stuff can add up to more total food than the big one, and yet the big one has much higher signal value. So we started setting up some of these vignettes where we would compare 1 to 1, and then 1 to 2, and 1 to 3, and so on. And the questions that Andrew would ask over and over again are things like, so here are 2 hunters. They just came back to camp at the end of the day. This guy got a deer, this guy got a peccary, and here's the question. Who would you prefer to marry if we're asking a woman, or if we're asking a man, who would you prefer your daughter to marry? Who would you prefer to have as your ally? And then I'm just going to jump to number 6 because this has been really important in the biological literature. This is called costly signaling in biology. Disease resistance. If sickness comes to the community, which one of these 2 guys is most likely to get sick? Because there's a belief that people, by signaling strength and health, are actually signaling to potential mates and allies that they have disease-resistant genes. So anyways, we build this giant chart where we have the rankings of how hard to find, how hard to kill, mate value, who gets chosen as the most preferred mate, most preferred ally, who has the highest disease resistance, and so on. And you can look at these relationships, and the first thing we discover is that size is a really strong predictor. but also to some extent hard to kill predicts on the y-axis here is mate value. So size predicts who you would like to marry or who you wish your daughter would marry, but also hard to kill items are preferred as mates as well. But we jumped immediately to the multiple comparisons where we could get big differences. And we did this in two ways. One is we set up comparisons where the total weight was equal. So one item versus three items. In this case, the total biomass is exactly equal. So if you were only interested in food, you should be indifferent to whether or not they get this one or this one. On the other hand, the ranking of what's harder to kill, jaguars are ranked way, way higher than deer, and therefore if you're interested in signal value, you'd expect people to always prefer the man who hunted jaguars. And then we ran the preference test again. So this is hard to kill as a function of mate value, where on the x-axis we have bigger and bigger differences in the signal between group A and group B, and this is the 50th percentile. What's important about that is that if you were indifferent, half the time you should choose group A, and half the time you should choose group B here. That's what should happen because these are exactly the same amount of food. But in fact, what you can see is there was a really strong preference across all conditions to always choose only group B. There's nobody who goes over a 50% preference for A, which means essentially that they're ignoring signal value and paying much more attention. They prefer several small game that are an equal amount of food over one spectacular high signal game. And then we did the same thing over a 15-week period, and we got pretty much the same result, which is that you could say, who do you prefer, hunter A that kills each one of these big animals and occasionally gets nothing, or hunter B that gets lots and lots of little animals that add up to about the same as the big ones? And here we got a spectacular difference. So up at the top, this measures the two groups, how hard they are to find, how hard they are to kill, and down here is the informant preference. 97% of the men and women we interviewed preferred many small game over few large game. So even if signal value is way, way higher for the large game, they basically are interested. Our bottom line conclusion here is that Aceh men and women prefer provisioners who provide food, not show-offs who do spectacular signaling. Now, am I running out of time here?
Yeah, you're just about.
Okay, so what I'm not going to talk about today is the third project, which is looking at food transfers. I only want to mention that we have this really cool project in Mindanao, southern Philippines, with a group of people called the Sea Gypsies who live out in houses built out over the ocean. Some of them live out of their boats. And we've been monitoring resource flows for the last 3 years in extreme detail with 2 research assistants. And all I want to show you is this, that we're looking at total production per day as a function of age and sex categories and how much they transfer out and how much they get back from other families, and doing a detailed economic flow analysis. And the big picture we're getting out of this is that you can see this really well right here. Let's look at women's production. The total amount they're producing per day is less than what they're sharing out per day. In other words, how can this be true that on average, women are sharing, I've transferred this into US dollar value, but we're measuring in food value. Women are giving away $1.60 worth of resources per day, but they're only producing $1.20. That seems like it's impossible. How can you be giving away more than you produce? And the answer is that they're getting gifts back in from other people of $2 a day, and of what they're getting transferred to them, they're then turning around and giving it away immediately so that they can actually give away more per day than what they're actually producing. The big picture in all of this research so far is that the level of economic interdependency in these marine sea gypsies is absolutely spectacular. The amount of flows of goods and resources is beyond anything that we had expected to see. So let me go to the last slide and I'll finish. So to understand social behavior, cooperation, cultural patterns, and so on, we first have to understand economic behavior, which we believe to a great extent flows from ecological constraints. So that requires detailed monitoring of resource availability, profitability, goals, patterns of transfer between houses and communities, and then theoretical models about what would happen if any of these things were likely to change. And I want to thank, again, the John Templeton Foundation for funding. Thank you.
Thank you, Kim. Fascinating paper for those of you in the audience who wondered what it would be like being a hunter-gatherer and what females would think of you if you were not such a good hunter, but a flashy one. The real details quantitatively of how economics drives so much of this organization in hunter-gatherer groups, and to spend I can't imagine how many hours Kim has spent with these dyadic questioning sessions and so on, collecting these data, but certainly allow us from an energetic point of view and a social behavioral point of view to understand hunter-gatherer societies better than we have before. It's my pleasure now to introduce Joan Silk. You see cooperation going up here. Her husband's helping set up her computer. Joan got her PhD at the University of California at Davis and joined the Institute a number of years ago after teaching at UCLA in Los Angeles. And she is focused primarily on questions that explicitly link studies of non-human primates to humans. She's especially intrigued in how natural selection shapes the evolution of social behavior in primates, including ourselves. So to this end, Joan has spent a great deal of time in comparative study in the structure and function of social bonds in 4 different species of baboons. But today, she's going to talk to us about the development of, interesting title, development of pro-social preferences, or how children learn to be nice.
People are always supposed to say that they're glad to be here, but in my case, this is absolutely and genuinely true. We arrived at ASU about 5 years ago after a long and happy tenure at UCLA, and moving late in your career is sometimes a sort of odd thing to do, but it has been a real pleasure to move here to the Valley and to become part of the IHO family. It's been a really wonderful move, and we've hugely enjoyed working with our colleagues here. So today I want to talk to you about some work which I've been able to do with support from the Templeton Foundation. And as I describe this work to you, I'm going to build up to where our Templeton project begins. And like a lot of the other work we heard about today, the work that we're doing as part of this project builds on a body of work that's been accumulated over time and provides us, provides us with ideas that we wanted to expand and amplify, and we've been able to do that with this project. So, as many people have mentioned today, one of the central things that motivates so many of us in IHO is understanding how we came to be and how, and in particular, how we came to be such exceptional organisms. We have language, we have art, we have religion, we have sophisticated technology, we practice agriculture, we've managed to domesticate animals, which I think are domesticating us. And compared to most other species on the planet, and certainly most other primates, we are also spectacularly cooperative. And as we'll hear about more later on today, cooperation plays a central part in human societies. So how are we cooperative? We're cooperative in many, many ways. We share food, for example. Food sharing is a universal feature of human societies. And food sharing is also representative of the huge amount of economic interdependence that we see within families and within groups in human societies. We are altruistic. We voluntarily make sacrifices that benefit others that we may not know. We're able to organize cooperation in very large groups and at very large scale, again, something which other primates cannot do. We develop social institutions, and in particular, we develop social institutions that are related to social order and justice. And the cooperation, we understand that the cooperation that we see in human societies is supported by a whole system of moral norms about how people should behave, and also cultural expectations about what happens to people who transgress against these social norms. And today I want to talk about one virtue of modern humans, generosity. And in particular, generosity that extends beyond immediate family and our small circle of friends. And I'm going to talk about the question, and when I wrote the title, I said how kids learn to be nice, but really how I think the question really is how kids become nice or generous or virtuous. So for someone like me who's used to counting things and not really used to watching people at all, I look for a way of describing behavior in a systematic way. So it's not really enough to say that we're generous. I want to know how generous we are, and I want to be able to quantify that so that I can test hypotheses and do comparisons. So how generous are we? Well, one way to answer this question is to borrow some techniques from a field called behavioral economics, which is a field of economics which is really attempting to measure human behavior in an economic context. And so one of the techniques that they've developed is called the dictator game. So let's play the dictator game. Let's imagine that I give each of you $50, real money. You can walk out of the room with it. But I'm also going to give you the opportunity to put some amount of that $50 in an envelope. And then I'll distribute the money in that envelope to someone else. You won't know who they are. They won't have an opportunity to thank you. You'll never get to know who they are. You'll never get a chance. You're not going to play the game again tomorrow and let them give money back to you. So, out of your hands, out of your life. The question is, you've got $50 in one hand. How much money do you put in the envelope? Think on it for a minute. I want you to have a number in your head. Okay, so it turns out that economists use this technique, plus a number of others, to measure how generous people are. And economists ran this game in many, many places, in developed Western places, which means I asked undergraduates what they do, college students. But Joe Henrich, who was a student of Rob Boyd's at UCLA, had the bright idea that he should run the dictator game in a small-scale society in which he worked. And this inspiration eventually led to a series of large-scale cross-cultural projects in which people played economic games all around the world. And they realized it was important to move out of the economist's comfort zone and really explore the variation that we know, that anthropologists at least know exists across the world. So here are the results from the dictator game. And this is the fraction of the stake that people gave away in the dictator game. Okay, so at the top, it turns out, are people from Missouri, all right, who give away almost 50% of that original endowment. And you can see that there's a lot of variation in how much money people give away in the dictator game in different societies. And I think the astonishing thing is it's above zero, right? People are giving money away to other people. This is a pretty good measure of generosity. So I have actually run a similar version of this game with chimpanzees because I was interested in the origins of generosity, the evolutionary origins. But today I want to, and you could ask me about that later if you should want to, but I want to focus on work we've done with kids because I was interested in how kids become generous. And there are really kind of two different answers to this. One is that they're selfish, but we teach them to be nice. And another is that as kids get older and they mature, they develop preferences that lead them to become generous. So we're really interested in how this develops. And so with Bailey House and Joe Henrich and a few other people, we developed a cross-cultural project in which we systematically assessed how generous children were in a bunch of different places, including a number of small-scale societies. And the results of this project, which Bailey House, who you see here, who's now a postdoc here at ASU, a lot of the work, this work is due to Bailey's hard work at the computer, learning fancy statistics. So what we did is we ran a simplified version of the dictator game with 326 children aged 3 to 14 years in these 6 societies. And here's the problem we posed that we gave to the kids. Okay, the kids are the actors. And with the children, a child is presented with a choice, 2 options. You can choose one option in which there'll be 1 goldfish cracker for you and 1 goldfish cracker for another child, or a situation in which there'll be 2 goldfish crackers for you and none for the other child. Okay, this is a discrete version of the dictator game, and we wanted to know what children would do. So what's important about this is the generous choice here is costly to the child. They give up 1 goldfish cracker. And although 1 goldfish cracker may not seem very much, these kids really like goldfish crackers. So. Okay, these are the results, which look like colored spaghetti, but I want to walk you through them step by step because it's really an amazing set of results. So let's just look at part of it at first. Each color represents the behavior of children in a different society. For your reference, the American kids are in red. And from the ages of about 3 or 4 to about 6 or 7, you see a decline in how generous kids are. So it looks like the youngest kids are pretty happy they're fairly generous. But as they get older, they seem to become more and more interested in having more goldfish crackers for themselves. Okay, so there's that. But then something really interesting happens. So somewhere between the ages of 6 and 8 or 6 and 9, what a period that developmental psychologists call middle childhood. Something happens. So before they're about 6 or 7, it doesn't really matter where the kids come from in terms of their behavior. The kids in all the different places were behaving more or less the same. If you add society to the model, your statistical model, you don't explain any more of the variation. That's the way of saying it just doesn't matter. But during middle childhood, the patterns in different societies begin to diverge. And where kids come from is really important in predicting how they'll behave. And what's really interesting is that kids begin to converge the behavior of adults in their own societies. So we also played this game with adults, and it turns out adults vary, and their variation in our game maps onto their variation in the normal dictator game. So kids in middle childhood begin to diverge, and they begin to head toward the behavior of adults in their own societies. So what's going on in middle childhood? We weren't really sure. But in middle childhood, kids are getting older, they're getting a little bit more independent, they're getting out of the house more, and somehow they seem to be getting more sensitive to the social norms within their own societies. So that's what their behavior suggests. So this is both an observation, an interpretation of the data, and it became a hypothesis. And this hypothesis is something that Bailey set out to test in his postdoctoral project at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. And Bailey set up a really interesting experiment. So he tested kids in Leipzig. And what this little girl is looking at, she's looking at the computer, and what she's seeing on the computer is a model who is saying one of 3 things. So one version of the video says the right one to choose is 3-3. The kids have a choice between 3 for them and 3 for someone else, or 5 for them and 1 for someone else. Okay, so an even generous distribution or a more selfish distribution. So in one video they hear the right one to choose is 3-3. In another video they hear the right one to choose is 5-1. And in a what's meant to be a control video, they hear you can choose 3-3 or you can choose 5-1. So if the change in behavior that we saw in our cross-cultural experiment is a result of children becoming more sensitive to social norms, then we reason that these kinds of instructional videos should shift kids' behavior. Actually, this is Bailey. I'm using the royal we here. This is actually Bailey's work, not mine. But anyway, so the result that Bailey got for the kids in Leipzig is that sure enough, you can shift these preferences around. Hearing someone say this is the right thing to do affects what kids do, and it seems to be really effective right in that sweet spot of middle childhood. So we can make kids more generous, we can make kids more selfish by providing them with explicit information about what the right thing to do is. So this supports the hypothesis that children in middle childhood are becoming more sensitive to social information about social norms. So then the next step is to ask whether or not the patterns that Bailey observed in Leipzig are also patterns that we see in other societies. Does it have the same impact? Is the impact effective at the same ages? And so we have launched a cross-cultural expansion of this project with support from the Templeton Foundation. And we've designed a study in which we aim to do two things. One is to replicate the effects of prompts or moral guidance in shifting whether or not that influences children's behavior, to assess what age kids are most sensitive to this kind of normative information, and to find out whether or not these patterns exist in other places. So far, this is a continuing project. So far, we've collected data here in Phoenix, and this is definitely the royal we, because I have not collected these data myself. Bailey and his research assistants have done it. And we've set people off in various places. We had a great workshop in January where everybody came. We all practiced the game. We all exposed our preferences and played with these lovely meeples that we ordered off the internet. And this is the project. This is the question that we're presenting to the kids. Okay, so you're the subject, and you have a choice, like we saw before, between one reward for yourself, one reward for a recipient, an imaginary recipient, which is being represented by these adorable meeples. I ordered them myself from the internet. They're really cute. They come in all colors. Or two for you. We call these tokens, but as one of the little girls that Bailey tested insisted on calling them jewels. And the idea is the kids play with the tokens and then they exchange them for real rewards at the end of the experiment. Okay, so yeah, it will run. So here's one of the things. Thank you so much. Okay, so here's Deanna. Oops.
This paper is okay. It is okay to choose. This paper is also okay. This is also okay to choose.
So, okay, so the video is working on the screen. So she tells them which one's okay to choose. That's the control video. Here is the generous prompt.
This
Sorry.
paper is right. This is good to choose. This paper is wrong. This is bad to choose.
Okay. And she does the opposite for the selfish prime. So the children see one of these primes. This is a between subject design. They see one of these primes, and then they're given an opportunity to choose one of the two options.
Okay. So, here, take the headphones. Thank you. So, Aaron, is this paper right or wrong?
Wrong.
Okay. And is this paper right or wrong?
Right.
Okay.
Right, he says.
Alright. So, you choose this paper. How many tokens do you get?
2.
If you choose this one? And how many tokens does this person get?
1.
Huh? If you choose this paper, how many do you get?
2.
Huh? How many does this person get?
0.
Okay. And one more time. If you choose this paper, how many do you get?
1.
And how many does this person get?
0.
This person gets 0?
Yeah. No. 1.
Okay, perfect. Perfect. Okay, go ahead. You can choose paper.
Choose this.
Okay. All right. So these ones don't go to anybody. This person didn't get any. And now you can take these ones and put them in your bag.
So this work takes a lot of patience, but you can see that what's built into this are what we call comprehension tests, so the children know what the right answer is, and then we can score that to see whether, how that might influence their choices. And this is the way these data get collected. This is a particularly amenable subject. You're running these experiments, children have to go to the bathroom, there are fire drills, their friends walk by. So it's, I thought this work would be infinitely easier than watching baboons, but it turns out it's really not that much easier. It's really not that much easier. Okay, so the question then is whether or not the video primes that they see, whether or not they are effective. And overall, they do work reasonably well. So the black line represents what children choose when they're given the you may do A or you may do B, right? Where it's neutral. They're much more likely to be pro-social when they hear the pro-social prime, the you should be generous prime, and much less likely to be pro-social when they're given the selfish prime. But you can see that at some point, even the children that are given the selfish primes turn out to be generous. And we think that this is a result of the fact that they have other information about the world. They know what they've been told, and that at some point they no longer are willing to do what this weird woman on the video is telling them to do. But we do think that the prime is impacting kids, and although we don't know exactly, we can't time it exactly, this is the kind of data that we need to compare children growing up in different cultures to find out how they're responding to this kind of priming information, and to get an idea of their sensitivity to these social norms that they're exposed to. Another thing that we want to do in this project, which is again one step further than we'd gone before, was to think about how children become aware of and how they respond to transgressions by other people. Because we know this is an important part of cooperation in humans. So cooperation in humans is kind of a carrot and stick. On the one hand, we think that people are really motivated to be cooperative in many situations, and we really have pro-social preferences that lead us to prefer outcomes that benefit others. But those preferences are, tugging against those preferences are a certain amount of selfishness and a certain amount of temptation to defect. And so we're also interested in how children become aware of and how they respond to these kinds of transgressions against social rules by other parties that in interactions in which they're not directly involved. So we set up a situation in which our subject now will be told about a situation in which two other individuals are interacting in a similar situation, and our third-party actor has either been selfish or generous. Okay, and the subject then will be told about this event and will have an opportunity to give one of their tokens, their jewels, to have it taken away in order that a penalty is imposed on this third-party actor. So the question is, and in the trade this is called altruistic punishment, so that you incur a cost in order to impose a sanction on someone else. And this is a feature of human cooperative systems as well. And you'll probably hear more about this later on. Okay, so do children do this? And we wanted to find out if this is associated with children's prosociality, their own choices, and if it's influenced by the kind of norming information that the social priming information that we're giving them. Things are complicated here. And we haven't, this Bailey sadly stayed up all Thursday night to make this graph for me. So this is just extremely hot off the press. And at first glance, so we have 3 conditions here. Let me just tell you why there's 6 graphs, because there's 6 conditions. A child can hear a prosocial prime, she can hear a selfish prime, or she can hear the neutral baseline prime, and then she can behave selfishly or nicely herself. That's 6 combinations. And you see a lot of curves overlapping except for this one in the lower left-hand corner, so let's zoom in on that, because that seems to be where things are something interesting is emerging. So this is a case in which a child has heard a prosocial prime. The right thing to do is to choose 1, 1, and has actually chosen 1, 1. Then that child, when asked whether or not she would like to give up a jewel in order to punish someone who's chosen a selfish outcome, she says, yes, indeed. So she's much more likely to do that. So that increases the likelihood that she will punish. So this, again, is happening in middle childhood, and we are extremely interested in finding out, first of all, in replicating this pattern, secondly, in finding out whether or not children in other places behave in a similar way or not. So this work will all be completed over the next couple of months, and the data will be coming in. I don't know how they'll turn out. That's the joy and terror of doing empirical work. You don't know. But it's been extremely useful to have this opportunity to be able to do the field work so that we will be in a position to answer this question and to find out a little bit more about how children become generous, how children come to have the virtues that play an important role in their own societies, so that we know there's variation across societies in the way adults behave, and we're interested in what processes shape the development of these preferences as children mature. So what we're thinking is that generosity is shaped by social norms that vary across society in exactly what you should do in different social situations. That children start to pay attention and be influenced by these social norms sometime in middle childhood. And that these norms affect not only their own behavior when they're interacting with other people, but it may shape their responses to others who do or don't follow those same rules. So this is a step in understanding the development of these preferences in children. And when we finish this project, we'll know more than we did when we began. So thank you very much for your attention. And I thank, oh, wait, I forgot. Gratitude is another virtue. So I would like to acknowledge the John Templeton Foundation, who's made it possible for us to do this work. I'd like to thank my colleague, Sarah Matthew, and her postdoc, Carla Hanley, who helped us pilot some of this work at their study site in Kenya. I'd like to thank all the ethnographers who are going to help us conduct this work around the world and who helped us with our previous project. I'd like to thank all the staff of the IHO who helped make today possible and who've helped us with all of the nuts and bolts of running the logistics on this grant over the last couple of years. Thank you very much.
Thanks very much indeed, Joan. Rob Boyd is professor in the School of Human Evolution, Social Change, and affiliate of IHO. He's been a real pioneer and leader in the field of trying to understand cultural evolution. And specifically, his research in his books and articles focuses on the evolutionary psychology of the mechanisms that give rise to and influence human culture, and how these mechanisms interact with population dynamic processes to shape human cultural evolution. And today, he is going to talk about, I don't have your title here, what's that? Cultural evolution in the laboratory. Looks like you're talking about a different kind of animal. Thank you.
Thanks, Don. Am I live here? Yeah, that's good. So, one other way to think about human uniqueness is to think about humans as animals. 3 million or 4 million years ago, we were a more or less normal ape, maybe with a little peculiar way of getting around. Today, we live in giant cities. We cooperate in huge numbers. We have technology and all these things. We're an animal still, but an animal that has been transformed over the last 4 or 5 million years into a really different kind of creature. And what I want to talk about today is the role that cultural adaptation plays in this transformation. So humans, the first thing I want to convince you of is that humans adapt differently than other animals. And this plays a crucial role in why we've been so successful. So most animals, that's not working. Well, we'll do it the old-fashioned way here. So for most animals, many habitats means many species. So there are 2,200 species of rodents or so amongst the mammals, about half the species of mammals. And they live in a very, very wide range of habitats over the entire world. But different habitats produce different species. So you have Arctic rodents that are little spheres, and desert rodents that are long, skinny ones for thermoregulatory reasons, and giant capybaras in the Amazon, and so on and so on and so forth. Rodents are specialists. They're genetically evolved specialists, and that's typical of primates as well. The typical primate has an ecological range that's limited to a few habitats and maybe 1 or 2 continents. Humans are entirely different. Humans live in every terrestrial habitat in the world except Antarctica. They've done so for 10,000 years, more or less. One species occupies everything from the high Arctic to the South African bushland to the forests where the Ache make their living. So humans are more adaptable than other creatures. Humans are able to adapt to a wider range of environments more accurately than any other mammal. And that transformation has occurred in the last few million years of human evolution. Now, people often emphasize the role of tool making in our humanity, but lots of animals make tools, or artifacts anyway. Tools, most animals don't have hands, so it's a little tricky if you define tools as things that you carry around. But many animals make very complex artifacts. So these are the nests of a group of African birds, the weaver birds, African and Asian. And they're superbly designed houses to raise your kids in if you're a weaver bird. And each species of weaver bird makes a characteristic nest that's adapted to its particular situation, its particular habitat. Humans also make nests, and if you look at the upper left-hand corner there, that's a temporary house made by Hadza foragers in East Africa. Seems to me that it's roughly the same level of technological complexity as the weaver bird houses. But the thing is, humans can make many different kind of houses. Tents for Central Arabia, gears for Mongolia, snow houses in the Arctic, And that house in the tree is for people who live in Irian Jaya and are extremely nervous about their neighbors. Imagine carrying the water up to your house every day. Anyway, so the thing that makes, one of the things, it's not the only thing, but one of the things that makes humans different than other animals is that we are able to culturally evolve highly refined adaptations to a wide range of habitats. There's no other animal that can do that. Now, how is that possible? Why is it that we're better able to adapt to a wider range of environments than other creatures? The standard answer is, and somewhat self-congratulatory answer is, we're just smarter than all those other creatures. We understand, we're cognitively more sophisticated, we've evolved more cognitive sophistication, and I think that's undeniably true. Humans are better at causal reasoning and theory of mind and a bunch of other tasks that seem related to adaptation, and that's the explanation. And I want to spend a few minutes to convince you that that's not the whole story. Don, what time am I starting at? It's 20 after now. So how long have I been going, I guess is the question. 4 minutes. Okay, I'm good. What I want to convince you of is we may be smarter than other creatures, but that's not enough to explain our ability to adapt to such a wide range of environments. I think there's a lot of evidence that this is the case, but I just want to focus on one of them, which is what I call the Lost European Explorer Experiment. The Lost European Explorer Experiment has been repeated many times over the last few centuries, in which a group of European explorers go off someplace, things go wrong, they run out of food, and they starve to death or otherwise can't make a living, in a place where people, hunter-gatherers or other people, have been living for thousands of years, raising their kids, and so on. So let me give you an example. In 1860, the Worthies in the city of Melbourne organized an expedition to explore the interior of Australia. So no European-descended people had made it across Australia going north to south. So they organized this expedition. It was led by an ex-military man named Burke, and a surveyor named Wills, and they set off with about 25 people and camels and firearms and oak dining room tables to eat their dinner on and all kinds of Victorian impedimenta, and things went badly wrong. Most of the expedition never made it. Burke and Wills and two other guys set off across Australia. They made it to the Gulf of Carpentaria, but on the way back they ran out of supplies, and in a place called Cooper's Creek, 3 of the 4 of them died, and the 4th one was found by a group of Aboriginal Australians and kept alive until the rescue expedition came from Melbourne a year later. And these weren't newbies, right? These were not urban people. Especially Wills had spent a lot of time in the outback, as they say, and knew a lot about getting along, and yet he couldn't make a living in this environment. And in particular, they got hungry, and they saw the Yandrawandra, who were the local Aborigines, eating cakes made from a local aquatic fern called nardoo, and it turns out that nardoo is toxic, and it's likely that Burke and Wills died as a combination of starvation and toxicity from this enzyme that's in nardoo. So they didn't die because they were stupid. They're just as smart as you and me and everybody else in the room, just as smart as the Yandrawandra. It's not brains that allowed the Yandrawandra to live in this place, raise their kids, be successful. It's the fact that they were heir to a rich body of cultural information, culturally transmitted acquired information about how to make a living in this place. that nardoo was toxic, that you had to wash it for a long time in water to extract this enzyme to make it edible. How to make cordage, to make nets. They got a lot of their calories from fishing in the local ephemeral ponds. And so on and so on and so forth. Individual brains, we're plenty smart, but none of us are smart enough to figure out how to make a living, how to do all the things that hunter-gatherers in different parts of the world do to stay alive. And what I want to talk about today is 4 really bodies of work aimed at understanding how this process works. The first one I'm going to tell you about is an unfinished field project, and then I want to tell you about 3 lab projects which have been finished, which shows you that field work is, in fact, harder than lab work, even if watching baboons isn't that hard. Anyway, so I want to use this to, I'm going to address 2 questions. The first one, okay, so there's this pool of cultural information. You're an Aboriginal kid. You're growing up in near Cooper's Creek, and you learn how to process nardoo, to detoxify it. Why is that there? How do people know that? If people aren't learning stuff on their own, what makes this pool of cultural information adaptive? And I think there's a kind of continuum of views about the answer to this question. At one end of the continuum, there are people who think that culture is like a library, that it's a store of useful information, and people pick and choose from that library because they understand what things work and what things don't work. So on this view, it's still really cognition that is the key to understanding human adaptation, but cognition aided by the ability to store a large pool of information that has been acquired by previous people in previous generations. At the other end of the continuum, we have what Dan Dennett calls competence without comprehension. At this end of the continuum, people are evolved, have an evolved psychology, which causes them to be predisposed to believe what people are doing around them is the right adaptive thing to do. And most people have no idea whether what they're doing is the best thing to do or some other thing to do. It's just what we do around here. On this model, cultural adaptation occurs because over longer time periods, various processes cause adaptive behavior to accumulate. For example, we know from experiments that people are predisposed to imitate the successful experiments and fieldwork, in fact. That will gradually cause adaptive behaviors to accumulate in a way that's quite similar to natural selection, even if nobody ever understands why the behavior is adaptive. Another process that can do it is if occasional lucky breaks or experts inject adaptive information that most people then imitate. And this is an important, which end of the continuum people are on is important for understanding other questions. So Joan just talked about little kids absorbing norms. And we know little kids are avid imitators. If you're at the end of the continuum where everybody is picking and choosing based on what they know what's good for them, then we shouldn't expect odd or maladaptive or other kinds of behaviors to evolve because it's still our evolved cognition that's picking and choosing what's right. If, on the other hand, we're at the end of the continuum where most people don't know what they're doing, then we can explain the cultural evolution of behaviors that are either cooperative and therefore individually maladaptive, or just plain maladaptive, of which there are many examples, I think, in cultural variation. But I don't want to go to that place now. But just to say that this issue, which end of this continuum we're on, matters. Okay, so first, the field project. So Kay showed us pictures of floods and 110-degree heat and stuff. Well, this is where I've done my field work. It's in Fiji. The temperature hardly ever varies from 80 degrees. And the main problem is not getting invited to eat breakfast 3 times or 4 times a day every day you go around in the village. This is a long-term field site that Joe Henrik started and which I've worked at several times. And I've been interested in the whether, so it's a place that still runs on well, it's changing, but when we first went there especially, it was a subsistence economy. 80% of the calories came from the garden or the ocean. And people built these traditional houses, used other kinds of traditional technology. And I got interested in whether people understood why they were doing the things they were doing, and I want to use as an example the design of these houses. These are called bure in Fijian. They're have lots of important functions. Keep the rain off, give you some privacy, keep the sun off in certain times of the day. And they have a lot of design features, and I focused on the strength of the building in hurricanes. So this part of Fiji, the eastern part of, western part of Fiji is called Hurricane Alley, and typhoons sweep through, and in the last 4 years, there's been 2 hurricanes which have destroyed about 40% of the houses in the village. So here's a cutaway of the interior structure of a bure. These posts go into the ground, there are rafters that go up, and there's this interesting structural thing in the middle. The outside is thatched. So and you can see this truss in the roof. There's a lot of structural features that seem to be relevant to whether the bure will stand up in the hurricane or not. And so 4 years ago, I started a pilot project where I interviewed the 8 most respected builders in the village about trying to elicit what they knew about the structure. And then with the Templeton grant, Michelle Klein, who was my postdoc and has worked in this village for quite some years, returned and did a widespread survey of trying to elicit whether people understood why the houses had the structure they had. At one end of the continuum, builders should be able to tell you the function of every piece, and at the other end of the continuum, no. And we didn't want to prompt them. If you ask people in this kind of situation, you know, what happens, why do you do this? Why do you put that big long post in the middle of the house? The thing's called the Vata Umbulavu. Can I point? Yeah, this long, very heavy beam has to be cut down in the forest and hauled to the house. Why do you put that? People find it difficult to answer those kinds of questions. So instead, what we did was we made drawings of houses that they don't build. So this house, for example, has many fewer posts, half as many posts as the standard house. And then we ask the builder, what happens to this house in the hurricane? Because if they know why they're doing what they're doing, they should be able to tell you what happens when you change something, and we don't ask them whether it's better or worse, but they will volunteer that quite easily. And in this case, no problem. They tell you, well, it's not going to be as strong, the wind's going to push the house over. On the other hand, this drawing, we've removed this long beam that goes down the center, the Vata Umbulavu, and now this piece here, which holds up the roof pole, is now resting on this other crossways beam. This is something that exists in houses throughout the Pacific. I don't know why it's there. I can't figure out the physics. The Fijians don't seem to know either. So they give no consistent answer about what happens. Now this project's not finished. Michelle's returning this summer to finish the survey. And when she gets back, I hope we'll have a better, more complete answer to, I should say, so it's not the case that Fijians don't know anything about the houses they build. They seem to have clear causal explanations in some cases and none in other cases. One of the problems with this project was I haven't been able to find any engineers who can tell me what that thing is for and a bunch of other features in the house. These houses are too different from modern houses for standard engineering wisdom to tell us what the right thing to do is. So this summer, we're starting a second version of this. Jake Harris, who you've heard about several times, is returning to work on the Hadza, and we're going to ask the same kinds of questions about bow making amongst the Hadza. So these are Hadza hunters. They still use bows they make on their own for hunting. Unlike the houses, we know a lot about the physics of bows, self-bows like this that are made out of a single piece of wood. Jake is, in fact, an avid bowyer and knows a lot about bows himself, and I think this will add quite a bit more information because we will be able to tell whether what the Hadza are telling us is correct. Okay, I've got 10 minutes, right? In addition to this field experiment, another postdoc, Maxime Durex, and I have been trying to test these same ideas in the laboratory. And what I mean here is we bring students, ASU students, into a computer lab with multiple computer stations. They learn a task, and then they're able to culturally transmit their solutions to that task to other students in the lab. And then we can manipulate, for example, so what you'd really like is a very unethical experiment. You'd like to take away the social influence from the Hadza or the Fijians and find out if they still can make the houses or the bows they make. And obviously that's not an experiment that's ever going to get by the IRB, but you can run that in the lab. And so Max, this is again, I wouldn't say the royal we, this is the supervisorial we. Max and I ran a series of experiments to test this idea. So we give students a task. The task is to get this box open. They're given 4 keys, which are these squares that have 4 colored sides to them, and they're asked to put these in those slots in order to open the box. And this person chose that and got the wrong answer, didn't open. There's only one of the possible arrangements that opens the box. They're told ahead of time that there's a rule, that if they can learn the rule, they'll be able to open the box every time. So here's an example of a placement that does open the box. And here's the rule. You have to select, this one has to have the same color as this one. The second one has to have the same color as the top, the third one of the right, the fourth one is the left. We intentionally chose an abstract problem so that we could eliminate any outside knowledge that people would bring to the problem. Okay, and now we ran 2 different treatments. We brought a whole bunch of students into the lab. Either they played as isolated individuals or they played as networked individuals, and they did this. So in the individual learning experiment, they had 40 time periods, 40 trials, to open the box as many times as they could. They got paid for how many times they opened it, and they couldn't see what the other people had done. In the social learning treatment, For 30 trials, they were in a group of 4, and they got to see what the other person did, whether they could open or not the box or not. And then we take away the social information, and they have to play 10 rounds on their own. And they know all about this in advance. So they know that this is going to happen, and then we ask them to play. So here is what happens in the individual treatment. People gradually learn how to open the box. So after 40 trials, maybe 30% of the people, on 30% of the trials, they're getting into the box. This is the network. So this is evidence for the fact that social information allows people to solve problems that they couldn't as individuals. The network people, by the time you get to 30 trials, more than 80% of the boxes are getting opened on each trial. Now the question is, what have they learned? Have they learned the rule as a consequence? Are they picking and choosing the rule by observing others? Or are they just copying others and mainly not knowing what they're doing? Well, the answer, you take away the social information, and they do almost as badly as the individual learners. But we can, so why don't they go all the way down? What's going on? And the answer is there are a small fraction of people that are figuring out the right answer. And once they get it, they always get it. Everybody else is just copying. So in this population, there are a few people who are experts that are injecting adaptive information into, whoops, that's the wrong one more. So most of these people are just copying without knowing what they're doing. Okay, the second set of experiments I want to look at is the effect of population size. So Kim talked about social network size being larger on the coast than in inland populations. There are a lot of theoretical reasons and some empirical reasons to think that population size affects the extent to which cultures can accumulate adaptive complexity. This is a bathymetric map of Tasmania. The only thing you're supposed to be able to see here is that there's some shallow water that connects Tasmania to Australia. During the Pleistocene, sea levels were lower, and Australia was connected to the Pleistocene, and people got to Tasmania, and when the Bass Strait filled up, they were isolated, and the technological complexity of their cultures diminished substantially between 10,000 years ago and 5,000 years ago. That's just an anecdote, so to speak, an N of 1. Michelle Klein and I collected data from databases on the complexity of marine foraging tools in Pacific Islands, and we found, where am I? We found a strong relationship between population size, so the horizontal axis here is log population size, and the vertical axis is a number of tools, and actually log number of tools, and you can see that there's a strong positive relationship, about as strong as it could be with only 10 data points. And 4 out of the 5 low contact societies, meaning smaller social networks, less contact with other islands, had lower than expected tool complexity, and the high contact societies the worst. So Max and I wanted to test this experimentally, and we published a paper in Nature Communications a couple of years ago. The first of our papers from the Templeton funding. And this was a problem. This is actually the first experiment we did, and here we used problems that, a task that made use of people's general knowledge. In the end, we regretted that decision, but people were given resources. They could combine the resources to make tools, and then they could combine the tools and the resources to make logs, and then they could use the logs to make totem poles, and they got paid for the complexity of their totem pole. So here you can see there were 27 new items they could make. That led to 115 different logs that they could assemble into 200 and some odd thousand totem poles, and the more complex ones produced higher payoffs. And the question was, in one treatment, people were grouped in groups of 6, another treatment, groups of 3, and in a third treatment, they had to solve the problem themselves. And the question was, how would population size affect solutions to the problem? And the answer is bigger populations produce bigger, better solutions, higher payoffs, they're more likely to produce a totem, all the measures that you could measure. And this number was, the mean here is higher than the very best person in the solitary treatment. So larger populations allow more complex cultural evolution. The only tricky bit is this is a graph in which there were groups of 6, but they were only partly connected. They didn't get to see all the solutions of all the other players. And you can see, now this difference isn't significant, but they're just a little bit bigger than the complete connected ones. And that's not the same as what the theory predicts, or had predicted, or what we saw in the data from Oceanian tools. So Max especially got interested in trying to understand why partial connectivity might lead to more cultural evolution. And we did some experiments, and it got published in PNAS last year. And basically, the task is different. I don't have time to explain the task. But the essential difference between it and the original task is now there are many different optima. And once you go down the path towards one optima, it makes it harder to get to the other one. And so the idea is now that if you split the population into partially isolated groups, different groups explore different parts of the adaptive topography. They evolve towards different solutions. And then when they're brought back into contact with each other, they can recombine and get to better solutions than a large group, which would go off towards one optimum and never get there. So we had 2 treatments, one in which groups of 6 stayed together for however many it is, 72 time periods. In the second problem, the groups of 6 were broken up into groups of 2. They were just worked as groups of 2 for a while. And then after 36 trials, they were migrants, moved from one group to another, carried their ideas with them. They could be observed by the subjects in the other group, and then they'd be isolated again, and then a different migration event would take place, and so on. And what's cool is we get this, I think, very interesting result. So the black dots are the rate at which the single group of 6 solves the problem. And notice they adapt more rapidly at first. They have more brains, they're exploring more territory, and they can evolve more quickly. But they get stuck at one of the optima. The population, which is divided into 3 different groups who only make contact occasionally, adapts more slowly, but in the end gets farther because it can explore a larger part of the space of solutions to this problem. Okay, so that's the work we've done. Thanks to Max Durex, Michelle Klein, and of course, the Templeton Foundation. Thanks very much.
In terms of innovation, in terms of invention, in terms of transference of breakthroughs, and has a big bearing, I think, on things that Bill Kimball talked about this morning in terms of Stonetools and so on. They might have been invented many, many different times in many different places by quite disparate groups of people. And if that particular group of individuals that developed the use of those tools went extinct, it might have disappeared until some other group invented it. So that there are applications of this sort of documented work to understanding perhaps early, it has early implications for early hominid behavior such as tool use and tool making. Good. So Sarah is an assistant professor in the School of Human Evolution and Social Change. Her work, her research explores the evolution of humans' ultra-sociality and the role of culture in enabling that. And she's especially interested in how humans evolved the capacity to cooperate with millions of genetically unrelated individuals. We, in crafting, in Bill's crafting of the Templeton grant application, we pointed out that there were a number of things that are part of really what make us human, and cooperation, as you have heard throughout today, is very important. And why is it that we are the only vertebrate on the planet that actually cooperates with everybody else who's a member of our species, as opposed to, say, our closest living relatives like chimpanzees? If you evolve and are born and are raised in Troop A that lives in Cave Creek, and there's another troop down here in Tempe that's in Troop B, we do not particularly appreciate it when chimps from there come into our troop, and very often they're killed and or eaten. So what is it about human behavior that is so unique, that makes us so cooperative, even though we are genetically and behaviorally and culturally unrelated to one another? And how this links to the origins of moral sentiments, pro-social behavior, norms, and large-scale warfare. And Sarah's talk today is entitled, "Is the Cultural Boundary Also the Moral Boundary? A Cross-Cultural Test of Cultural Group Selection." Thank you.
So I have a bit of a laryngitis, so that's why I want to keep my microphone at the highest possible volume. But it's late in the day, and I thought I would start with a story, because I think this is a pretty heavy topic. Is the cultural boundary also the moral boundary? So this predates the Templeton Foundation grant and goes back about 10 years to a time that I started doing my dissertation work. And for this work, I went to a population in northern Kenya called the Turkana. They are a pastoral group of subsistence herders. They are, like many pastoralists in this area, making a living herding cattle, camel, sheep, goats, and donkey. And they are limited by access to dry seas and water wells, and because of this, they have to periodically, they live in temporary settlements, and due to that, they have to periodically relocate in search of new pastures. The Turkana also participate in cattle raids against neighboring groups. So these are raids in which they mobilize in order to go and attack another group, get cows, fight over territory, fight over water wells. And as part of a larger study on warfare that I did among the Turkana, one of the things that I did was ask people from this particular territory of the Turkana called the Kwatela a particular set of questions. So this was part of a vignette study in which I told the Kwatela about Kwatela warriors who would go off to raid animals from the neighboring tribe. So this is a story in which Kwatela warriors go off to raid animals from the Toposa, the tribe across the ethno-linguistic boundary. And I also did a very similar study in which I told them about a group, about warriors who go and raid animals from another Turkana territory called the Lukmong. And in both cases, the warriors go and raid animals from these neighboring groups, and they bring cattle back to the Kwatela, the subjects of the study. And so the Kwatela are basically benefiting fairly equally in both these situations, but they had very, very different reactions to these two scenarios, to the warriors in these two scenarios. So about the warriors who go and raid another Turkana territory, the Lukumong, the bars in red are showing you how they reacted to these warriors. And basically, they think these are really bad people. They think that what these warriors did was wrong. They want these warriors to be criticized. They want to punish these warriors. They don't want to stand next to these warriors on a raid. They don't want to entrust their animals with these warriors. They don't want to lend these warriors an animal if they ever need help in the future. They don't want to let their daughters marry these warriors. Yet, they had completely the opposite reaction to the warriors who go and raid animals from the neighboring tribe, the Toposa. So those are the bars that you see in blue. Almost nobody thinks it's wrong to do that. In fact, they were filled with praise for those men who went off to the neighboring tribe, raided animals, and brought them back to the Cuartello. So what's really interesting about the situation is that the Cuartello would benefit equally from both of these scenarios. In both these scenarios, they get animals. And the Cuartello themselves are a pretty large group of people. They're about 100,000 people. But the scale at which they cooperate, the scale of their moral concerns, seem to extend beyond that of the Cuartello. It seems to also include this group of people called the Lukumong, but it doesn't include everyone. Clearly, it doesn't include the Toposa. It's okay, they think, to go and raid animals from them as long as the Cuartello are benefiting from it. And so that is, in some ways, one of the most important empirical questions that's going to tell us about the origins of human cooperation. What is the boundary of our moral concerns? Why is it that this group of people, the Cuartello, have moral concerns that extend to include the Lukumong, but don't extend to include the Toposa? So you might think that, well, maybe it's the ethno-linguistic boundary, it's the cultural boundary that's driving this, but why is it? Why should it be the ethno-linguistic boundary? Why should evolution, cultural evolution, whatever biological evolution, have shaped human pro-social psychology to operate at this particular scale? So in order to explore that, I will divert a little bit back into the basic question of why cooperation itself is a bit of an evolutionary puzzle. So cooperation is basically, the kinds of cooperation that we're interested in explaining is cooperation in which an individual does something that is at a cost to themselves and benefits another individual. And what happens is that individuals who just get the benefits and don't pay the costs are going to do better than the individuals who pay the cost and also get the benefits. So cooperators will always have a lower fitness than free riders or defectors who are not paying the cost of helping other individuals but are receiving the benefits. And as a result of this, biological evolutionary processes seldom produce cooperative behavior except under very specific circumstances. So this is a diagram showing you how a defector, how defectors would essentially, because they have higher fitness than cooperators, increase in frequency in a group. But it's also clear that groups of cooperators can do better than groups of defectors. So cooperators are creating these benefits for the group. So at the group level, cooperation can be useful. And if only selection can act at the level of groups, you can get cooperation to evolve. So if you imagine a system like this, where in blue you have groups of cooperators, red are groups of defectors, then over time, groups of cooperators would proliferate and the population can be dominated by cooperators. But the reason why this can't work is that in order for selection to act at the group level, you need to have variation at the group level. And what happens when you have gene flow, when individuals disperse and migrate into other groups, you have gene flow, and that means that you can start off in a world in which there's quite a bit of variation between groups. So here you have a group filled with cooperators, a group filled with defectors, but individuals, when they migrate, are going to erode that between group variation, so that over time, you're not going to have much variation preserved between groups. But humans have culture, as you've heard about for a good part of today's symposium. And the question is, can culture fundamentally change this, this dynamic or this constraint? And it turns out that it can. So when individuals have culture, suppose the cooperative behavior is a behavior that you culturally learn from other individuals, from observing the action of other individuals, then you can actually have individuals migrate from one group to the other. But when they migrate, they switch their behavior because they look around them, they see what's the locally adapted thing to do, who are the successful individuals in this population, and they change their behavior. And so you can actually maintain a fairly high level of between-group variation, even with high levels of migration between groups, if the behavior, the cooperative behavior, or any behavior, is learned culturally as opposed to specified through our genes. So in some ways, so this theory, this process is called cultural group selection. It was an idea that was put forward by Robert Boyd, who you just heard from, and Pete Richardson, his close collaborator. And in some ways, it's a pretty major theoretical breakthrough in understanding how human cooperation can evolve, because it basically alleviates one of the biggest constraints that has prevented the evolution of cooperation in many biological systems. So it's a great idea, but is it true? And any idea can work in theory, but we do need empirical tests of the predictions that these theories generate. And two really key predictions of cultural group selection. One is that between group variation in cultural traits should be higher than between group variation in genetic traits. So this is essentially the reason why we think culture alleviates this problem and selection can act at the level of cultural groups. So the question, the test is it true that cultural variation between groups is higher than genetic variation between groups? And second, a key part of the story is that the scale at which cultural variation is maintained is also going to be the scale at which cooperation should occur, the scale within which humans should cooperate. So in the project that we did that was funded by the Templeton Foundation, one of the key goals was to be able to test these predictions in a pretty comprehensive and compelling manner. And so I was fortunate to be able to recruit a postdoc, Carla Handley, to join this project. Carla had done field work for 10 years in 3 different pastoral societies in northern Kenya, not just one. And these were the Rendile, the Samburu, the Borana, and the Rendile population. And combined with the Turkana, who I had been working with for the last 10 years, we now were really well situated to be able to conduct a cross-cultural test of the predictions of cultural group selection. So the first part of our work was to measure cultural variation. So we're trying to estimate something called the FST, which is going to tell us the proportion of the total variation in a population that lies at the group level. The higher the FST, the more variation occurs between groups, the more the possibility and potential for cultural group selection to operate. And this is something that people use in genetics quite a bit. So genetic FSTs is basically the total allelic variation at a locus that is accounted for by between group differences, and we're using the analogous concept to measure the cultural differences. And so cultural FST is the total variation in cultural traits or a set of traits that's accounted for by between group differences. And there have been very little previous studies of exploring this issue. So there are two studies that have explored cultural FST values and tried to estimate them. One of them was done using variation in song characteristics in 16 Austronesian-speaking populations. Another study looked at variation in folk tales across 31 modern European populations. But a key problem in this work is that it doesn't it doesn't track individual level behavioral variation, because songs and folk tales are fundamentally a group level characteristic. So you can't really easily estimate how much of the variation is between groups versus between individuals. And there is one study that comes close to what we're trying to do. This is a study that used World Values Survey data and looked at 154 pairs of neighboring countries to look at the level of variation between individuals and between groups in people's values and beliefs and ideas. But the focus of this study was on industrialized Western populations. The group in this study is the country. And because countries have top-down rules, governments, laws, etc., there's lots of reasons why people will vary in their values between countries. And two other gaps, while all of these studies actually measured cultural FST, none of them tested the second part of this prediction, which is a very important part. Does the cultural FST help you predict the scale of human cooperation? And in order to do that, we need to have data on multiple social scales. So we need to have social groups that are nested within larger social groups so that we can see whether people are cooperating at a social scale in which the cultural variation occurs. So this is, we were well situated to address this because we have now 4 ethno-linguistic groups when we combined our forces, the Turkana, Borana, Rendille, and Samburu. And within these groups, we sampled individuals from multiple clans. So these ethno-linguistic groups are comprised of various different clans, and clans are very important social groupings. So we sampled people from 2 clans, 2 Samburu clans, 2 Borana clans, 2 Rendille clans, and 3 Turkana clans. So we have data from 750 individuals who span these 9 different clans spread across 4 ethno-linguistic groups. And to measure cultural variation, we collected data on how people vary in their beliefs and norms in a wide variety of domains. So we used 50 different norms and asked people whether they agree or disagree with these norms in order to assess cultural variation between individuals and between groups. And in order to measure the scale of cooperation, we conducted 16 vignette experiments that used culturally relevant contexts of cooperation, and we tried to understand to what extent is cooperation, people's likelihood of helping, going to vary if the target of their help is either a member of their clan, a member of a different clan, or a member of a different ethnic group. So how does cooperation change in these different contexts as the social scale changes from clan to outside of clan to outside of ethno-linguistic group? And this was a pretty elaborate project. So all of the data collection for this project was spearheaded and conducted by Carla Handley, and with the help of several RAs. So Carla did about 3 field seasons just for this project, which was only one of the 3 projects that we did as part of the Templeton funded grant. And it required about a dozen RAs at least working over a period of 12 months. So in some ways, this is, we understood why such a study had not been done before. In the process of doing this, it's something that was really only made possible by a large grant that could allow us to hire field assistants, a large number of field assistants, and be in the field for a pretty long time. And so, after all this work, I'm going to summarize the results in 2 graphs. This is the first graph. So this is showing you the FST estimates that we have, the cultural FST estimates. And below the blue line is the genetic FST values that we've taken from the literature, the known genetic FSTs between human populations. Above the blue line are the cultural FST values that came from these previous studies that I just described to you. And the colored bars are the cultural FST values that we've found from this data collection. And the main thing to note is that the cultural FST values are much bigger than the genetic FST values. So the largest genetic FST value is 0.07 in the literature that we could find, but the largest cultural FST value is about 3 times that, so 0.21. And some of the largest FST values come in when you look at the differences between ethno-linguistic groups, not the differences between clans within an ethno-linguistic group. So the red bars over here are the cultural FST values between ethno-linguistic groups. The blue and the green bars are the FST values between clans within these ethno-linguistic groups. So if we look at this, we've addressed one part of our prediction, which is that the cultural FST values do provide a much bigger scope for group selection to operate on compared to genetic FSTs, and we should expect the bulk of the action to be between ethno-linguistic groups, not between clans. So, and just a side note, the current genetic FST estimates we have is from the literature, but we're also actually in the process of estimating that for these populations on our own. So we have 500 cheek swab samples in order to do the genetic FST analysis from these populations, and we're collaborating with Ann Stone and Melissa Cyrus-Wilson's lab here at ASU in order to estimate that. And the last part is whether cultural FST is going to predict the extent to which people help. And that's basically the part of the prediction that hasn't been tested at all in the literature and is a really key part of testing this theory. So cultural FSTs giving you the between group differences. And the prediction is that as cultural FST increases, that means groups then become very culturally variable, the level of cooperation across those group boundaries should decrease, because cultural group selection is going to create cooperative norms that make people cooperate within the cultural group. And that is exactly what we are finding from this data. So this is showing you the regression of the proportion of helping an individual across a social group, and on the x-axis as a function of the cultural FSTs between those groups. And as cultural FST increases, proportion of helping decreases, and it explains a very big amount of the variation that we see in this data. So it's consistent with cultural group selection, and it's a pretty big effect. So in some ways, this is the effect that has been alluded to several times during the day, is basically the effect of culture in shaping other aspects of human sociality, including, and in this particular case, human cooperation. So in some ways, what we have here is a situation in which culture evolved for something else. So the talk that you just heard from Rob is getting at the crux of why humans are a cultural species in the first place. It's because culture confers us with adaptive knowledge that allows us to live in a variety of different habitats and build technology that no single individual can possibly build. But a really important consequence of the evolution of that capacity for culture was this, this effect that it has on the levels of between-group variation in human populations. And so it's not just that by being cultural, we became cooperative. It's that by being cultural, you got cultural group selection to happen, and that has allowed humans to evolve the capacity to cooperate. And the fact that it happened through that particular process has pretty big implications for the scale of our cooperative behavior and moral considerations. So we are cooperating well beyond the scale of close genetic relatives and people that we know. So we're super cooperators, but we don't cooperate with everyone. The fact that cooperation was shaped by this process of cultural group selection means that our moral sentiments are parochial, culturally parochial. Our moral concerns, to a large extent, are going to extend to individuals within our cultural group, and not so much to individuals outside of this cultural group. So thank you to the Templeton Foundation for allowing us to test this hypothesis that has long been waiting to be tested. And part of this work was also funded by a grant from the Carnegie Corporation of New York. Thanks.
Can we have the house lights up, please? And thank you, Sarah, yet again for another interesting paper and presentation. All right, I'd like to thank all of you for coming this afternoon and this morning, and I hope you'll join me in thanking our speakers who certainly enriched our day. Thanks very much.